Okay, I'll see you.
See you. Hello, good morning everyone. So, welcome back to the Gian program, Next Generation Sustainable Alternative Proteins for Food, Fundamentals and Technological Innovation. We are on the day 5 today. So, today we are going to have 2 lectures. The first one is nutritional evaluation of alternative proteins with a special focus on how do we evaluate amino acid profiles or composition, digestibility of a given protein and including their bioavailability. And in the later section, the lecture number 11, we are going to talk about allergenicity and anti-nutritional factors associated with the alternative proteins. So, these are the 2 lectures we are going to have today. Okay. So, let us begin with the first lecture, that is a nutritional evaluation of alternative proteins, mainly amino acids, composition, digestibility and bioavailability. So, today we are on the day 5 and let me try to summarize what we have been discussing over the days. Let us see where we are today with respect to our alternative or understanding with respect to alternative proteins. We began with the greenhouse gas emission, remember? That was our day 1. We began with greenhouse gas emission and we thought that okay, it is nice to have alternative proteins, right. And on the day 2, what did we discuss? Extraction, right. So, we discussed more about okay, there is a lot of different types of cells are present and we need to extract proteins to get them as ingredients, so that we can create new texture and so on, right. And the day 3, what did we do? Yes, we have extracted proteins now and we really wanted to know what kind of proteins we have extracted. So, we did characterization, that was our day 3, where we investigate or studied about FTIR, STS page, LCMS, chromatography and many things we have discussed. And we talked about primary structure, secondary structure and all these things and their importance, right. And the yesterday was day 4? Yes. So, yes, yesterday was day 4, we understood the properties, especially the functional properties, right. We talked about water holding capacity, gelation, no not gelation we did not cover unfortunately, foaming and all these things we discussed, and we also discussed one more topic, emulsification, and these things we discussed, right, properties. And we also discussed about how to create structure using extrusion, for example, right, shear cell technology and so on. Now, if you look into all these boxes here, we are progressing nicely, right. We started with the problem and we found a solution. Now, we are understanding that solution, right. The solution is alternative proteins, let us say, And now slowly we started how to get alternative proteins. Then we understood what did we extract by characterizing them. Then we understood, okay, we have extracted something and we have characterized something. Are they ready for food? Are they functional? That we have evaluated. Today we take the next step, that is digestibility and probably a little bit about health. I am not going to too much, right. Now we have alternative food protein product in our hand, right. Everything is good. We have characterized it. We know the functionality. It is tasting good. It's smelling good. No problem. That's not the end of the story. We have to eat it, right. Now we are starting eating it. Then we ask a lot of questions, right. What are the questions when it comes according to you when you are supposed to eat a new product? Yeah. Yeah, give her a mic please behind. Yeah. What are the question you think? I am giving you a new product, okay. I tell you, oh, this is having a fantastic texture, flavor is good. Now you have to eat it, right. What are the questions comes into your brain when you want to eat a new product? Not idli, dosa, not jalebi, new one, okay.
Taste.
Taste, one you think is like, okay, taste. Now imagine I told you already it tastes good, many people have told it tastes so nice. What else you think?
The composition.
Composition.
Because what are the components present in that particular stuff will be more of importance, ki how much protein it is giving and what are the amino acid profile.
Yes, she is thinking about composition. That is very important for me. I would always ask, like I would turn the package and see, oh, what is the nutritional value of this product? What is the calorie amount I am taking because of by eating 100 grams of this product? How much carbs it has? How much minerals does it have? How many ingredients does it have? How did they produce? Is there any label on that one? Do they say vegan, vegetarian, or less processed? Any labeling is there? Is there any FSSSI seal? All these things I will try to look for, because now I am a scientist and I have little bit knowledge in food area. I really want to look for all these things. Do you have any point?
Ingredients.
So she also thinks about composition.
But the additives they are used,
exactly.
The preservatives,
she
they are harmful for us or not.
Exactly, and she thinks now she has a lot of understanding now about the proteins. Now you guys are studying food science and technology. You know little bit about formation of good and mod molecules. You know little bit about some ingredients are good, some ingredients we have no idea whether good or bad. So you try to think about that. Anything else? She, yeah.
Allergens and toxicity.
Absolutely, this is an important topic, allergens, because many people are allergic to some sort of food or food products or food ingredients, right. And if you look into, for example, just take an example of a Maggi packet, right. Just turn it out and see somewhere they write something in bold. Have you seen? Some ingredients they especially write it in bold. Sesame, it's in bold. Why? Because some people are allergic to certain kind of food ingredients or food components and they develop allergies. I will talk about that also later. It is a very important thing that you have to look. If you are allergic to some food, some kind of ingredient, very important, do not directly eat that novel food there people are giving to you. Look for the composition and see. That is something we would like to have in our mind. Is there any allergic compounds, right? Anything, another allergens in other words. Any other things you want to see? Yeah, she has a point. All the way to the back, the mic will go all the way to back.
Appearance.
What does that mean?
Appearance means how the product is looking. Is it good or not?
Okay, the appearance of the the appearance of the product, that is also very important. Yeah, I agree with you. Good point. And we have another person there. Yeah.
Manufacture and expiry date.
Yeah, exactly. Manufacturing and expiry dating. That's super good point, right? When it was manufactured and when it will be expired. It something talks about the stability of the product, right? Some products are stable for only a certain period of time. After that, they may degrade chemically or they become harmful microbiological point of view. Maybe bacteria will grow, maybe yeast will grow, maybe some unwanted mycotoxins will be released. So, we have to be careful with about manufacturing date and expiry date, right. These are the things definitely we would look. I personally will also look for the price, how much is costing, right. What I am eating, whether it is matching with the price I am paying for it, right. That is also an important thing. We can't give a hefty price for every food product. So, these are things we will also. Apart from this, me as a scientist, one thing you completely forgot is something about is digestibility. Now, she mentioned about, okay, there is a lot of composition I will look for. Oh, now it has 10% protein. Oh, I am very happy. Protein, it is 10% protein. It has a very small amount of fat, but it has a very good amount of PUFA, polyunsaturated fatty acids. It has minimum ingredients, everything looking good. But 10% protein, what is present in the product, may not be so good proteins. Do you agree? It is not about amount. Now, we are going from amount to the quality aspects. Please understand that one. When you look into the label, when they say it has 25% of protein, they are not necessarily, I would like to use the word necessarily, a good quality protein, right. Right. So, there are proteins in some other things. If you eat it, then maybe they are not coming to your body, just maybe you are excreting it off, then probably they are not an important protein on a nutritional point of view. Does that make sense? So, now we are in the battle between quantity and quality. Most of the industries look for quantity because that is very important for them, because they want to sell it to many places. I do not think they do not worry about quality, they do. But we as a scientist, we as a consumer, we should think both quantity and as well as quality. Does that make sense? One of the important quality parameter is digestibility, and we are going to discuss that little bit later. Now, before going to the protein quality parameters, again I would like to refresh your memory. I will come back with this figure again and again and again. Okay. Quality is nothing but is again functionality was related to protein structure. Quality is also related to protein structure and its composition, right. We talk about high quality protein, what does that mean? Which is more bioavailable, right. The protein which is more bioavailable, we think it is a high quality protein. Anything else? What do you mean by high quality protein? Providing all the essential amino acids. Now, she is talking about two things, essential amino acids. We discussed that also. I will come back to another slide about that one. The composition is very important. What kind of amino acids does a protein contain, and that is something really important to determine the quality, because we need to consume essential amino acids, right. Then protein quality is again related to different levels of food protein structures, right. It is not just a amino acid composition, how the protein is bending, how protein structure look like this one, what kind of folding does it have, how it is interacting with some other molecule, is it having an association with other compounds, all this determines the protein quality, right. If it is having a very rigid structure, imagine if a protein has a very rigid structure, it does not want to open up even after you put it into your stomach, then that protein probably not very bioavailable, may not be very digestible, it might be some excreted off, right. The product might say it has 25% protein, but that protein is not available for me, it is only in the product, then it will go in feces, right. So, that is the reason why you need to be a little bit careful about when we say amount, right. It is more about the quality now. So, the protein structure does matter about determining the protein quality. Okay. Now, I also taught you the plant cells and microalgal cells in the second day, right. We mentioned to you, we discussed that in case of plant cells, maybe the proteins are associated with next to the endoplasmic reticulum, right, and there are some kind of protein bodies and probably they are accumulating proteins. And in case of microalgal cells, we have protein perhaps around in the chlorophylls, right. And we mentioned about one important aspect, what is that? The outer thing, cell wall, right. And we know that cell wall is a wall, it is a wall for the protein, right. So, proteins are inside now, cell wall is outside. So, if you eat, for example, microalgae as whole, though proteins are of high quality, it has a very good essential amino acid composition, maybe that protein is not available for us. We are not talking about the bioaccessibility and bioavailability, right. So, basically the body does not have cellulase, enzymes and other carbohydrates. So, it has a very high amount of protein, but we can't use it in the body, right. So, we have to keep it in mind. The quality is also something to do with where exactly protein is present in the given food system, okay. And that is the reason why sometimes we can't eat raw food. You get stomach bloating and other things, right. You get indigestion when you eat some raw food, because the protein is not digestible, it just wants to go away. Make sense? Oh, this we can, and in case of seeds, seed is having a fantastic source of proteins, right. 35%, 20 to 35% proteins can be accumulated in seeds, extremely good source of seed. We of course eat rajma, moong and other things. Do we eat them raw? No, right. We do not eat them raw, because if you eat raw, your mother says, do not do it that one, it is not digestible, you get dysentery or something like that, right. She always says that when you are small, do not eat raw. Some things can be eaten raw, not everything. Some soft tissues can be eaten raw, but not these hard ones, right. Why? Because cooking helps to disintegrate the cells, cooking helps to release the protein, cooking helps to unfold the protein, cooking also kills some unwanted things present in this along with proteins, and that improves the digestibility. So, we human beings made our digestive system in such a way that most of our food has to be cooked, not all, most of them we eat cooked food, right. That is the reason why, because the protein and other things are more accessible when they are cooked. Make sense? Now, I will ask you try to discuss why do we have to eat proteins, right. I have also told you in the very first class, proteins are important, it gives you energy. 1/3 of our calorie intake should come from proteins for the better development of our body, mind and everything. So, now I am not a nutritional or a health specialist, just to give a basic information here. So, we get proteins for energy, right. Once you eat proteins, there will be digestion, which I will explain in detail later. Once it is digested, we get amino acids or small peptides and so on, right. And then there is something called body amino acid pool. It is not a real pool, but there is imagine on a theoretical ground, there will be pool of an amino acid, right. Now, this amino acid will be utilized by the body to synthesize new protein, right. So, you digest it, you get amino acid in the intestine, and that goes to the blood. There is something called amino acid pool everywhere in the body, right. Now, that amino acid becomes a building block for another protein, right. You ate a protein, right, you ate a protein and it becomes amino acid, and this amino acid in the body becomes another protein. It is just cycling things, right. And some part of amino acid is also utilized for some other purpose. For example, DNA synthesis, RNA synthesis, they all DNA and RNA has nitrogen, right. Sometimes that nitrogen comes from the nitrogen of amino acid, right. So, some portion will also used for some other synthesis. Now, what I wanted to tell you here is that we need some continuous supply of amino acid to the body, right, because every day you are growing, right, every day you are growing. So, we need a continuous supply of amino acid, and it is said that I think we apparently dismantle about 250 grams of proteins per day, though you do not eat that much, you eat around 60 to 70 grams of protein per day for the. What does it mean? That it is a dynamic process. The proteins what we eat will be metabolized, and body also tries to metabolize the protein which is already present in the body. For example, in your muscle tissue, right. So, you have a supply, but they also try to eat their own proteins. The body tries to eat the protein already present in the body, especially in the muscle, let us say. Now, if you eat less and less proteins, what happens? Exactly, there is no source coming, body wants protein, but we are not eating enough protein. So, the body will try to disintegrate the protein in the body and that is what we say, you became lean. If you did not eat too much food, you look very lean, right? We say that, you know, you look unhealthy, right? Because if you do not eat food, you become unhealthy, right? Because the body is so dynamic, it is so active, it wants to work every single day, even though you want to sleep, but there is a lot of metabolism, they want to work continuously, and that they start disintegrating our own body proteins, and that might lead to health effects, disorders, disease and something. But we need to find out a healthy balance between how much we eat, how much body should disintegrate. I am not telling that body should not disintegrate muscles, it should do it. It's a normal process, but there should be a fine balance between the amount we eat and the amount of protein that body disintegrates every day. Does that make sense? Now you know why we have to eat the proteins every day? Yes? Cool. Now I have a protein bar or I have a protein containing meat analog, I ate that one, right? We have been talking about protein. One thing, the take home message is that body cannot use proteins directly. You know that, right? So when you eat protein, it comes to stomach, then it goes to small intestine, and the protein does not cross the intestinal barrier to go to the blood. Anything we eat, at the end of the day, it should go to somewhere, blood or gut or somewhere, right, to become more available. But the protein is so big, right? We are talking about the molecular weight 15,000 Dalton, 300,000 Dalton, 400,000 Dalton, right? And these are a very big protein compared to sugar. Sugar is how many Dalton? 200, 300 Dalton or 400 Dalton. It's very small molecules, right? So the body cannot transport, cannot utilize usually the bigger molecules. There are some exceptions, of course, because we do not have something called diffusion phenomenon and active transport phenomenon, especially if you have large big molecules, because the body has actually a gate wall, right? You have food here, you have blood here, and to cross the food from here to here, there is a barrier, right? Now proteins are so big, so they are not directly bioavailable. To the best of my knowledge, none of the protein can go into the blood to be nutritionally available. So now, since the protein is not bioavailable, the protein has to be digested in the body, and that is called digestibility. That is the fraction of proteins that becomes bioavailable, and that is called digestibility. Am I clear? It's a very basic things, I'm pretty you already know, but I try to refresh your memory. And there is something called bioavailability. We always get confused with digestibility, bioavailability, bioaccessibility, bioactivity. There are many words here. They all go hand in hand, but not the same. Okay? Bioavailability means the rate or extent to which active ingredient, in this case amino acid, becomes available at the site of action. So that is called bioavailability. But don't get confused. For now, I will use the word digestibility and bioavailability every now and then. Basically, they are an associated terms, right? Digestibility is the amount of protein that becomes bioavailable or that becomes absorbed in the body. That is digestibility. Is it clear? All right. Now, I will just teach you protein digestibility in 1 or 2 minutes, because this is a 10 marks question in your high school, right? On BSc also, okay. In your BSc you studied digestibility, right? It's a 10 marks question, so we need to draw this nice human being figure and oesophagus and crazy head and intestine is lying here and there, and it was not an easy job for me. I was poor in drawing. Okay, now I'm going to teach you digestibility, how exactly it happens. I'm a human being, I have a food in my plate now, I take it. First thing I'll see is that I feel it, right? Is it anything digested in the hand or on the fork or spoon? Yes or no? Something digest? Yes, you might squeeze it little bit, but nothing is digested. Agree? You're just changing the texture. Do you take a rice? Maybe you might do it like this one, right? You mix it with curry. You take chapati and you soak it in rajma, right? And then the chapati becomes soft, right? But it's not digested. It is just changing the texture of the food. And now you take that piece of chapati with rajma and you put it into your mouth, right? Then what will you do? If you are in a hurry, you will just bloop like this one, because hostels we usually do that one, because I give only lunch break for half an hour and you are in a hurry and you do not have to do something just like this one. But the first thing is that you digest in your, I mean you not digestion, sorry, you chew in your mouth, right? And what happens, there is a lot of saliva that our salivary gland secretes, the watery substances, right? Whenever you think about jalebi, that watery substance comes out of the mouth, right? Especially children. When I say chocolate, it comes right in their saliva here, right? It's the body, such a, you don't have to eat something. Just by thinking the saliva will come out, right? That's the beauty of the human being's body, right? Even dogs do that, of course. Now, the saliva comes in your mouth into play, right? And then you start chewing, okay? Basically, you're grinding the food substance, a bigger particle into small particles. You might also break some cells because of this chewing process. And at that time, the body, especially the mouth, the salivary glands, and there are some glands here, they release an enzyme called amylases. Amylases are enzymes, they hydrolyze what? Starch, right? They like carbohydrate. So, the first step of digestion occurs in our mouth. But one important thing, whether protein will be digested in the mouth? No, because we don't release and did not release any protein degrading enzymes. We released only carbohydrate degrading enzyme called amylase. So, proteins are still intact in the mouth. Please note that degradation of carbohydrate is also important. Why? Because sometimes proteins are associated with carbohydrate, right? The cell wall is surrounded, right? But we do not have cellulase, but starch is surrounded the protein, for example. There is a complex interaction between protein and starch. Now, if you hydrolyze starch partially, the protein is more available. Does that make sense? That is the first step happens in the mouth. It's nothing is digested yet. Protein is not digested yet. Okay. Then it comes to the so-called stomach. That's an important organ we have in our body. That makes you hungry, right? So it comes to the stomach. In the stomach, there is some acid. Stomach releases an acid. What is that called? The stomach, this is stomach that releases an acid called HCl. It also releases some enzymes called pepsin and gastric lipases, right? Pepsin is a protease. Pepsin is a protease. What do you mean by protease? It can hydrolyze proteins, right? So basically, pepsin hydrolyzes the protein that is coming in the stomach. It will not completely degrade protein, it will just partially degrade protein. No, complete digestion will not happen in the stomach. Another point, partial digestion will happen in the protein. Nothing is absorbed now, nothing is absorbed in the body. A partial hydrolysis of protein has happened because of pepsin. Pepsin is an enzyme, it is active only in acidic condition, right? It is active in acidic condition. That is the reason why body releases acid. I think pH is around 2-ish, something like that in the stomach. So pepsin becomes super active, it cleaves the protein. Now the bigger protein become kind of a large molecular polypeptides. Okay. Now that will come to the small intestine, right? The next organ we have connected to the stomach, small intestine. The small intestine has a lot of enzymes, trypsin, chymotrypsin, carboxypeptidase. There are many enzymes are there, and they basically degrade a larger peptide into smaller peptides, smaller into much smaller peptides. All of the enzymes are acting one by one, right? At the end of the day, we usually get tripeptide, dipeptide and an amino acid. Am I clear? So that is the final fate of you, because the body can absorb 3 amino acid containing peptide, 2 amino acid containing peptide, or a single amino acid. If you have 4 amino acids joined together, then it is not available for the body. Am I correct? Am I clear? So it has to be digested, it has to be hydrolyzed in a way that there should be 3 amino acid maximum that the body can utilize. Is it clear? The digestibility. Now that peptide and free amino acid, this free amino acid, these are peptides, dipeptides or dipeptides. Now that becomes ready to go to the blood. Okay. There is a table here, I mean there is a different figures with the same meaning. So then there is a barrier here that is called let us say intestinal barrier, just to make it very simple, and there are some transporters. For example, there are some peptide transporters. These are again proteins. They carry these dipeptides and tripeptides on their head, and they will bring from outside to inside, from intestine to blood. Okay. Sometimes some amino acids can also have a diffusion based on sodium transportation, that is all nothing. But what I wanted to tell you here is that dipeptides and tripeptides and free amino acids can be transported from intestine to blood. I think some people say that usually peptides are more bioavailable than free amino acids, I am not very sure, but just remember that tri, di and free amino acids are transported to the bloodstream, and this is the basics of digestion happens in our body. Any question? If I make it a 10 marks question, will you write now? Yes, that is a Gian question for you to pass. Digestion, is it clear? What the take home message here is that there are lot of enzymes in the body, they are responsible for hydrolyzing the protein from a bulk into small particle, clear? From bulk size into small peptide. This is a table where different enzymes that body synthesizes for hydrolysis and what kind of conditions and so on. I do not teach all these things, is there on the PowerPoint slide. If you are really interested in, go through it, okay. It is not important for me now. Now the protein I was talking about quantity versus quality, and the quality matters, and one of the important thing for the quality is again as I mentioned amino acid composition, where we need to talk about essential amino acids, right. Essential amino acids are those the body cannot synthesize. There are 9 essential amino acids we need to consume every day, right. And it is not just about the quantity or the essential amino acid, it is also about their proportion is important. Imagine you have a protein with only a lot of lysine, nothing is present. Is it a good protein? No. Imagine your protein has a lot of every essential amino acid, but methionine is completely lacking. Is it a good protein? No. So the composition is also important. There should be fine proportion of these essential amino acid. Just remember this point, plant based proteins usually lack methionine, which is an essential amino acid, and some proteins also lack lysine, it is also very essential amino acid. Also tryptophan sometimes, okay. Also cysteine, cysteine is not essential, it is a conditional essential amino acid. These are the limiting amino acids in most of the plant based proteins, and that is the reason why plant proteins are not necessarily equivalent to dairy proteins or meat proteins. There are some amino acids present in a very high quantity, some are completely absent. Make sense? And then we should talk about digestibility. It is not just about the quantity and quality of essential amino acid, it is also about the digestibility that I have just discussed with you. How much is digested and bioavailable, that is also important. The digestion is again, this availability is again dependent on the chemical integrity. That means sometimes amino acids are present, but they are not present in the right form. They might be oxidized, okay. They may be associated with polyphenols, they may be associated with tannins, and sometimes they may be bioavailable, they may be immediately oxidized somewhere, and then they are also not good amino acids. So the chemical stability, their integrity is also important. Now these are the 2 main parameters affects the quality of proteins. Now do you understand the importance of quality of the proteins? Whenever now if you talk with your colleagues or family members, when they say high amount of protein, you should talk to them, okay, it's high, I agree with you, how about its quality? How about its essential amino acid content? How about its digestibility? Please note that today the food labeling, if you look into any package, what they just mention is amount of calorie, it is a mathematical calculation. They mention about protein content. Do they mention digestibility? Do they mention about essential amino acid content? No, we are not in that stage yet, right. So you as a scientist think a little bit beyond what is there on the package. Am I clear? Alright, this you already know, essential amino acids and conditionally essential amino acid. Now, a little bit move on to the measurement. Excuse me. Okay, the quality of the protein is determined by amino acid composition and digestibility or bioavailability. Okay, two things. This is what the summary of my previous slides. Now, I am going to explain you how are we going to evaluate amino acid composition of a given protein. What is the content of essential amino acid? What is the content of non-essential amino acid? That is what we are going to discuss now. Now, this is my protein, it is a big protein. Okay. Now, I would like to know how much essential amino acid is present in this one. I will give you 1 minute to think alone, and then you will tell me what are the strategies we can use. I will just give you 1 minute to think how I can evaluate the amount of essential amino acids present. Think back about my previous lectures, everything. Yeah. Oh, yeah.
And we can utilize.
I'll get back to me. I'll give them 30-second more. Then I'll get back to you just to think. Yeah, yeah. Three, 2, 1, 0. Okay, now.
So you mentioned about in our previous lectures, you taught about the Edward degradation,
degradation.
that we add that component and that component will be attached to the first terminal and that will be degraded, and further we can remove that component and we can assess that amino acid. So how we can this is how we can determine the sequence of the amino acid.
Exactly.
Else we can go for LCMS also, and there is a amino acid analyzer based on the as their amphoteric amorphoteric nature can also be that property can also be utilized in the determination of specific amino acids, as well as the presence of specific groups which are particular in certain amino acids only. On the basis of that analysis we can go for the determination of amino acid composition.
Super nice. Yeah. Any other, I'll get back to this point, any other technologies you're thinking about? How we can measure the essential amino acid? Did you think anything else? Okay, she was telling about our first method that we discussed where the degradation approach, you can cut the N-terminal amino acid one by one and measure, then you get the sequence of the amino acid. That's a good idea. And she also mentioned about every amino acid has a unique functional group, right? Somehow you attack and determine that unique functional group, then you can get an idea, and you can use LCMS to measure because that measures the mass of a given component. We can also use chromatography to measure amino acid composition. There is a lot of different approaches to measure amino acid composition, but I am going to teach you the most important one that we are using today to measure essential amino acid composition, a little bit more about analytical chemistry now. Is it okay? Now, the protein has individual amino acids like this one, right? Now, I have an essential amino acid here, and I have an essential amino acid here, and I have an essential amino acid here, imagine. Okay. I want to measure how much is lysine is present, how much tryptophan is present, how much methionine is present in the protein, in a given amount of protein, right. The easiest thing is, as she mentioned, find out a functional group and see what is, but it is very difficult in protein system. Why? Because protein is very complex first of all, they are folded and amino acids are present in different positions, and there is no good reagent that will selectively bind to the amino acid you want and give you the right data. There are some, but that is not available for every single amino acid we are talking about. That is not possible today. Okay? We need to quantify this in a very accurate way. I am using the word accurate, means we need to find out the true content of lysine. We just do not want to have an approximate 5%, no. We want to know the true content, exactly how much is present per gram of protein, right. That is what we need, but the protein is very complex and it is very difficult to measure the protein amino acid composition directly, very challenging. For that, but the easy thing is that if you run this protein to a chromatographic column, you get peaks. Remember my previous lecture? Proteins can be separated and you get the peaks, and peaks are directly proportional to concentration, and you can make calibration curve and you can find out the quantity. But proteins are very big and we can't, now amino acid is present inside the protein, they are attached. We can't measure that, we are measuring the protein but not the amino acid. Now what strategy I can use? First you have to unfold the protein. The first step she is saying, you unfold the protein, okay. Now I unfolded the protein become linear, imagine. Then I have injected to the HPLC, I got only one peak. It is not the amino acid peak, it is again a protein peak.
First we have to unfold the protein, then we have to further digest into small peptides.
Exactly, and she is telling that first you unfold it, now you convert into small peptides, right. And now I will inject to the column, then I will get multiple peaks, but they are associated to peptides.
Peptides only.
Okay,
yes.
But I want amino acid. What will you do now? Now again digest the peptide bond further. You convert every protein amino acid into individual amino acid. Okay, you convert every protein amino acid into individual amino acid. Now inject to the column, and then you get a lot of peaks, and each peak perhaps corresponding to individual amino acids. Is it clear? This is the base of amino acid analysis.
No, there is instrument named as amino acid analyzer.
Yes.
So that analyze on the basis of this
exactly. You need to first
only.
hydrolyze protein before you inject through the amino acid analyzer. I will talk about that one later.
Okay.
Yeah.
Okay.
Is it clear? The basis of amino acid analysis. If you inject protein directly, you do not get amino acid composition. It is difficult, very difficult. Then she said you do make a digestion, but you digestion, if you do partial digestion, you get peptides. It is also basis of peptide analysis you just mentioned. This part we do it in the peptide analysis, but we need an individual amino acids. You completely hydrolyze protein into, I use the word hydrolysis, protein into amino acids, and then do the chromatography to identify and quantify individual amino acids. Then you can quantify the amount of essential amino acid present, amount of non-essential amino acid present. Do you have a question?
No, I was, there is current research going on in the field of protein that more of the people are getting into the interest, or they are exploring the bioactive peptides.
Yes.
Because they have, I guess, the prebiotic or symbiotic properties,
yeah.
And they are being considered as nutraceutical
Yes.
of potential.
Yes, this is another side application.
So this is how we can go for the analysis of biopeptide also,
Absolutely.
means using HPLC.
Absolutely,
Right.
Exactly the same phenomenon.
Okay.
You can
Thank
do
you, sir.
intact protein analysis also. For example, I mentioned how do we measure cruciferin content in rapeseed, size exclusion chromatography. Directly inject protein. It talks about only protein, doesn't talk about amino acid. Now she wants to do the bioactive peptide analysis, because she can create bioactive peptides from protein by doing partial hydrolysis. How do we measure that one? Again chromatography. Inject to the peptides, you measure it. But we want individual amino acids, so you completely hydrolyze. It's called complete or total hydrolysis. Total hydrolysis. My handwriting is not so good. Is it okay? Is it clear? The approach we are using? Yeah.
Sir, can we really measure bioactive peptides by using chromatography?
Yes,
technique?
we can measure bioactive peptide using chromatography combined with MS.
Okay.
Okay, what we do here is that I don't cover all these things. I'll quickly answer your question. What we do is that some food already contains bioactive peptides because there are some, for example, fermented foods. There are some bacteria, they release some proteases, they convert big protein into small peptides, and they create bioactive peptides themselves. But you can artificially create bioactive peptides by adding enzymes, right? You can do that one. So once you get the bioactive peptides, I just use my clean slide here. So you had a big protein, okay, this is my big protein. The peptides are here, the bioactive, these are the bioactive peptides. If you use a bacteria, the bacteria will cleave these peptides and then give you the bioactive peptides, a lot of bioactive peptides, different ones, okay. You can also use enzymes to do this job, right. And then you inject these all peptides in a solution to HPLC, right, and the peptides will separate one by one and you get a peptide chromatogram like this one, right. If your detector is MS, it will talk about the sequence of these peptides, right. And when you know the sequence of the peptides, there are databases available. You know that there is a database called bioactive peptide database, there is a lot of ones. You compare your amino acid sequence with the database. If they are matching, they say, okay, this is maybe having anti-glycating property, maybe this peptide is having anti-inflammatory property and so many things. Okay, it is all depending on which composition they have, which combination, lysine, glycine, alanine and so on. So peptide analysis is also done like this one. It is not only for bioactive peptides, some peptides are giving flavor. For example, curcumin peptides, that gives you the meaty flavor, right. The small peptide, 3, 4, 5 small amino acid containing peptides, they give you the meatiness in the mouth. There is a lot of research going on in that direction also. How do we identify peptides that gives you a meaty flavor in your mouth? Again, they are analyzed in this way. Did I answer your question? Yeah, yeah.
You were telling about how to measure the amino acid, and we talked about there are different steps we have to carry out for the sample preparation. Is there any instrument which carries out all these steps means automatically to determine the amino acid, or do we have to do it manually, means at the laboratory scale to carry out step by step for the sample preparation, then we have to inject that in the particular instrument to get the amino acid composition?
That is a very good question. I will talk about 30 minutes about that one now.
Okay.
Okay? That is my next, exactly what you asked, I will answer now. Is it okay? Alright, so how do we do amino acid analysis? It has two major steps. Not everything is automated, many things are automated. There is no instrument in the world at the moment, automated instrument, that you take a protein and get the complete amino acid composition. Unfortunately, no, but people are developing on that one, but there is nothing at the moment. There are machines to determine alcohol content, there are machines to determine carbohydrate content without doing any sample preparation, but not amino acid individual at the moment. I am not telling research is not going on, it is going on, but we do not have anything yet in the market. So we have to do little bit manual work here, not a little bit, a lot, and I have spent around 6 years on these methods of my life. So it's a lot of work to be done to quantify individual amino acid. So amino acid analysis has two experimental parts. One is protein hydrolysis, the second one is chromatography. Okay, protein hydrolysis is nothing but, I mentioned to you, you have a lot of a big protein with lot of amino acids. The hydrolysis is nothing but breaking the peptide bond. Okay, hydrolysis is nothing but breaking the peptide bond and converting the protein into amino acids. Once you get the amino acid, then you go and inject to the column, and then you get a chromatogram containing individual amino acid as a peak, and the peak area is directly proportional to concentration, and you can plot a calibration curve and you will get the amino acid content. This is the basic approach we are using today, and this approach is also approved by WHO, FAO, ISO and all the organization which talks about quality parameters, they use the same method, same approach. Is it okay? FAO, you know, right, Food and Agricultural Organization, FTA, Federal Drug Administration Organization, even FSSI, ISO International Organization, all of them use the same method. Okay. All right, now I will talk a little bit more about protein hydrolysis. As I just mentioned, it is nothing but breaking the peptide bond. The chemistry is simple. Amino acids are joined one together by a bond called peptide bond. It is an amide bond. Can you see here? So, when you do the hydrolysis, you will cleave this bond here. Okay. Hydrolysis is nothing but cleaving in the presence of water molecule. Hydro means water. The water is an important ingredient that will cleave the peptide bond and get you individual amino acids. These are amino acid 1, it is amino acid 2. This is amino acid 1 bonded to amino acid 2 in this case, and you get the free amino acid. Can you see? And the process is called hydrolysis. But this is a very slow process. If you take a protein, put it into water, do you get amino acid? No, right? Proteins are always protein in the water. But if you store it for 20,000 years, maybe you will get free amino acids, because that reaction is so slow, right? They are energetically hindered. There is a big activation energy barrier. So, water cannot break peptide bond directly. Now, how do we help it? You can use enzyme. I will get back to you that one. What else we can use? We can use enzyme, not reducing agent. How was that? Temperature. So, you can heat. You know that by heating, rate of chemical reaction increases. That is organic chemistry they have taught you, right? Every 10-degree raise in the temperature, reaction rate is doubled. So, heat it, you can fasten the reaction. But in this case, even if you apply a lot of heat, peptide bonds are so stable, they do not degrade. You can use enzymes. Enzymes are what? Catalysts. Enzymes are catalysts, they increase the rate of chemical reaction. Can you give me an enzyme example that hydrolyzes the peptide bond? Pepsin, all the proteolytic enzymes you can use, okay. If you do not have enzymes, you can use acid, HCl you can use. You can also use base, NaOH. The proton and hydroxide ions can catalyze hydrolysis, and we use that one. We use acid, we use base, and we use enzymes to hydrolyze protein into individual amino acids, and I am going to talk about all the 3 techniques we have established in our lab now. Okay? We use enzymes, we use acid, we use base to cleave the peptide bond to get the free amino acids, and all the methods have their own advantages and disadvantages, and we will also discuss about that. Okay? And if there is any doubt, please raise your hand, do not hesitate to ask question. Okay? Now, let us see the acid hydrolysis. There is a protein with the peptide bond here, use acid and it will cleave the peptide bond into amino acids, and the condition we are using here is that 6 molar hydrochloric acid and temperature is about 110 degrees Celsius. She mentioned about temperature, and acid is a catalyst here, and temperature is 110 degrees Celsius, and we heat it for, can you see how long? Now 110 degrees Celsius. So, now you can imagine how stable is a peptide bond, right? We need to eat HCl, 6 molar is nothing but concentrated hydrochloric acid, 50% water. Is 1:1 diluted concentrated hydrochloric acid is 6 molar, right? Such a strong acid, plus 110 degrees Celsius, plus 18 hours, and then the peptide bond hydrolyzes. Now, you can imagine how stable a peptide bond is, right? But body digests that in our stomach because of the enzymes. Yeah.
There was one, I don't have any scientific reason on this, but many of the nutritionists were saying that the people who are taking whey protein concentrate for their muscle bodybuilding, the protein remains in the intestine for long duration.
Yeah.
So is this the reason that the acid which is in fact present in our body, that it's HCl only to degrade or digest the protein and complex food matrix, still the protein, if we are taking more than our RD, means daily allowance, so that protein can harm the body in fact.
In principle, yes, because the body creates X amount of HCl and Y amount of enzymes.
Right.
They have a limit. Now, if we eat too much protein, we say you get indigestion. It's simply because it has to retain for a particular period in your stomach. Then the promoter thinks, now you don't enough in my place, you just go out. Then it will push to the intestine. Intestine will release enzymes. Right. It has also have a limit. Then it will push to the gut.
Right.
And then the bacteria will start acting on the protein. And then you get fart and other things when you eat more and more protein, right? So it's more about the finding a good balance.
So
children cannot eat this much protein, they eat small. Bodybuilders eat a lot of protein because they are actually they are consuming protein so fast to build the muscle. It's more about how much you have and how much enzyme is released. You need to find a good balance. Yeah.
Just to add on to this, means excess of everything is bad, so excess of protein is also bad.
Yeah, of course. I think what is the disease called ketonuria, right?
Yes, that is due to the
ketonuria. So it's you don't eat too much protein, but that is very rare cases. We are not that rich in India or in developing country to eat excess of protein. That's the problem of some other people and some class of people, not everyone. All right. So is it clear? But this is happening artificially. I'm not talking about the stomach will have acid 6 molar, no. Stomach will never have 110 degrees Celsius, it's only 37 degrees Celsius, right? And the protein will not stay in stomach for 18 hours, no. In stomach, there is mostly because of enzymes. Enzymes are more clever than the acid to do the job. I'll come back to that later. Now, this is we are doing in a glass bottle in the lab. Is it okay? Are you understanding what I mean? This is an experimental approach to hydrolyze protein, but the same thing happens in the body, but in a different way. All right. So, and then we heat it at this long period. Please note that you need 18 to 24 hours to cleave the peptide bonds. But for some peptide bond, for example, if you have a peptide bond with valine and isoleucine, for example, you have to heat it at 110 degrees Celsius up to 7 days. So, they are super strong non-polar amino acids. Okay, you need to heat it for 7 days. So, we do most of our experiment for 24 hours in our laboratory, to 24 hours. Proteins are digested. So, we do it in a way that we take in a small system, we add acid, okay, and then we put nitrogen or we remove the oxygen from the bottle. Why do we do that one? To avoid oxidation. If there is oxygen is present, they will degrade the essential amino acids, some of them. So, we have to remove them. So, we remove oxygen or supply nitrogen, then we heat it in a oven for 110 or 24 at the individual bottle, what we call hydrolysis. But this is its own problem. When you do acid hydrolysis, cysteine is completely destroyed. You don't get any cysteine, completely destroyed almost. And tryptophan is also completely destroyed if you use HCl. And methionine and tyrosine over 5 to 2%. So, this method will not work if you want to identify every individual amino acid. Some of them are completely oxidized, some are partially degraded. Okay? But another problem is that if you have a glutamine, it becomes glutamic acid because you will hydrolyze this amino acid also. If you have asparagine in your protein, it becomes aspartic acid. These are the problems of this hydrolysis method. It is called artificial modification. We did not want that, we do not want that, but it happens. Is it clear? And then we have alkali hydrolysis where we use base 4 molar sodium hydroxide, same conditions, and this one we use especially to determine tryptophan, because in the previous method tryptophan is completely destroyed. Can you see? So, if you want tryptophan in your protein, you need to do base catalyzed hydrolysis. Often we do 2 hydrolysis for samples. one acid hydrolysis and one base hydrolysis. In acid hydrolysis you will get almost everything except tryptophan and cysteine. In base hydrolysis we will specially get tryptophan. Is it okay? And as I mentioned, these methods have its own problems. For example, you have a loss of degradation of tryptophan, tyrosine and methionine, and we in our laboratory we use an acid called methane sulfonic acid instead of hydrochloric acid. The beauty of this acid when you use for hydrolysis is that you get a better recovery for tryptophan, tyrosine and methionine. So, we do not have to do 2 times of hydrolysis for the same sample. We can do only one hydrolysis to get almost everything except cysteine. So, use methane sulfonic acid as a hydrolytic agent. You know, I mean those who are studying masters here, that you might think it is an overwhelming amount of information. However, for you the take home message is that think that there are different acids available to create protein hydrolysates, that to convert protein into individual amino acid. Just think that much is enough. I am also teaching for the online people here. There may be PhD students sitting there doing this analysis. It is a very important analysis we do in protein science. Okay. So, the challenge in this particular method is that, can you see, we wait for 24 hours up to. Time is money. PhD students are always in a hurry, right? Industries pay a lot of salary if the person has to wait for 24 hours, and of course, the person can do other job, but time is always money. We need to reduce the time. So, to do that, back in the days I developed a method using microwave. You know that microwave, kitchen microwave, right? When you boil water, when you make boiled water on a pan, it might take 10 minutes. The same temperature you can get it maybe in 5 minutes in microwave. Microwave, sorry, some water molecule absorbs microwave and they dissipate heat and become super hot very quickly. This method I was telling we need temperature, can you see? So, this heat we can generate quickly with microwave, right. So, we developed this microwave assisted methods. Now, it hydrolyzes proteins completely in 12 minutes. The previous one was taking 24 hours. So, we can reduce the hydrolysis by minutes to 12 minutes. There is a lot of difference, can you see? 24 hours is one full day, 12 minutes is just a coffee break, right. So, that is the method we are using in our laboratory today to hydrolyze proteins to get into free amino acids. Now, we also found that this method gives you a fantastic recovery with respect to when compared to other acid hydrolysis. The methionine recovery was really high in microwave to conventional method. Conventional recovery was around 40%, with microwave we can recover of methionine. That lower to a lower extent it happens in microwave compared to conventional method. As this is a nice table, I found it from this research article no review article. It clearly shows that some of the advantages and disadvantages of individual method you use for hydrolysis. You can see that in almost every acid and alkali hydrolysis method you degrade asparagine and glutamine, they become glutamic acid, unfortunately that is a common thing. And acid hydrolysis usually destroys methionine, okay. It will completely destroy cysteine. So, if you really need cysteine and methionine, you need to do another kind of hydrolysis, which I am not covering today. So, they are advantages. Do you have a question?
Sir, optimize this conditions for hydrolysis using what power duration used is?
Very good. Not mention all these things. I spent about a month kind of finding out every different conditions where I get the best recovery. So, this microwave is not based on the power. That is the bad way of doing microwave synthesis according to me, because the power in the microwave is measured by watt, right. In the kitchen microwave, you take a cup of, you want to reheat your coffee, for example. You have a kitchen microwave. You want to reheat coffee. You take a coffee and put it on that plate. The plate is rotating, you know, and you just turn the knob and write minutes or select the minutes. I need 2 minutes, right? And it heats nice. But if you keep the coffee cup in the center, the temperature in the coffee cup may be, let us say, 65 degrees Celsius after 2 minutes. Okay? If you just keep that coffee cup on the side, change the position little bit, the temperature may become 55 degrees Celsius or 70 degrees Celsius. This is the biggest challenge with the microwave with power based, because it is very difficult to optimize the temperature and the position of the tube that you place. That is a little bit challenge. It is not very reproducible. When we do chemistry, we need to be very accurate and precise and reproducible. When I do the experiment today, I should get the same result when I do tomorrow. So that reproducibility is very difficult to get. And also, if you have some other components present in this one, then the microwave phenomenon will change. So that is the reason why we do not do it in a power based method. This is a beautiful microwave instrument. It works purely on temperature. Of course, it applies microwave power, but it adjusts the temperature. That means, if you say I want 110 degrees Celsius, it will maintain 110 degrees Celsius by adjusting the power. Very, very reproducible temperature adjustment it will do. So it is called organic microwave synthesizer actually. So in our case, we use 150 degrees Celsius for 1 minute, followed by 165 degrees Celsius for 10 minutes. That is the hydrolysis method we are using.
Sir,
and I optimize it with so many permutation and combinations. I do not show that one. Yeah.
Sir, is this microwave same as we use in our kitchen?
No, no, it works on the same principle, but
not in terms of this application, but in terms of frequency, like in home kitchen.
Yeah. Yes.
Like 250 or 950 megahertz.
Yes, it works.
Is the frequency same in this scheme?
Yes, it is still more power kitchen microwave. A lot of parameters you can control. You can control the pressure inside the vial, you can control the temperature inside the vial, you can also have a stirrer to rotate, you stir the sample while it is heating. There is a lot of options you have in this instrument. It also, kitchen microwave can cost you, let us say, 5,000 rupees. This might be 100-fold high expensive than that one. That is the difference.
Yes. Sir, one more question is that,
yeah.
In case of microwave heating, mostly the heat is generated from inside, volumetric heating.
Yes.
Just in case of this hydrolysis, chemical hydrolysis, we apply heat from the outer side, from outer to inner.
Yes.
But in case of microwave, it is from inner to outer.
Yes.
So, is there any difference?
I mean, that volumetric effect could be very, I mean, it is important in microwave. It is a normal phenomenon. That is how you heat it. But we are not talking about big or this size basically. It calculates, let us say, of your something like that. Use only 3 amps. So that thing very mind, heating for one not 3-4 seconds, 1 minute and up to 10 minutes. That is, and plus we have a stirring, the homogenizing. So heat is equivalently very quickly. Second, I would say. So that is not a big challenge in our system.
Sir, one more question. Are we using same conditions like in acid concentration?
Yeah, concentration is the same. A little detail, but I just want to method. Concentration, 6 molar HCl we can use, and 4 molar methane sulfonic acid, all the same. It is only the time and temperatures are different. The time is maximum 12 minutes here, temperature I just mentioned, in that is 110 degrees Celsius.
Sir, what is the technical reason for the breakage of this peptide bond? In case of micro, it is so easy. But if we carry out this process in another traditional methods or any,
it's so basically, I mean, if I don't want to teach the phenomenon or principles of microwave technology here, we all know that creates heat so fast. Conventional heating. Conventional heating method is this. You have a fan, you put oven or water bath, right? the heat is going in this direction, what we call convection principle, right?
Conduction.
And it's a very slow process, it goes layer by layer by layer by layer, right? If there is no stirring, it's again a nightmare. And to heat the water bath, if you want to heat the water bath at 90 degrees Celsius, to heat the water bath itself, it will take half an hour more than that one, right? And then the heat to dissipate inside the sample, it will take another 20 minutes. So, to get a temperature 110 degrees Celsius inside, that will take a long time. So, one of the important thing. And the second thing is that we are 50, 65, but for a short duration. We change this chemical rate of chemical reaction much faster in microwave compared to this one. Microwave itself will not bring any chemical reaction. Am I clear? Microwave itself will not bring any chemical reaction. Microwave is very weak radiation, it cannot break the bond. It will be broken purely based on catalysis and HCl and acid and so on, but the heat is helping, that is it. Microwave radiation itself is not breaking the bond, do not keep it in mind. Microwave cannot bring chemical reaction, yeah.
Sir, in case of milk, we are using just ultra high temperature. We are using suppose 143 for few fractions of seconds.
Yeah.
Can we use similarly conditions for this method? Like if we suppose carry out same process using 5 in ultra high processing techniques.
So you mean can we use microwave for pasteurization?
Can we replace microwave with another any technique, like in case of milk we are using ultra high temperature treatments?
For the hydrolysis?
Yeah.
Oh no, you can in principle use it, but who will use a UHT system for acid?
For
steel
scale.
and everything?
No.
Don't do that. That is, we have a proverb in Kannada, that means ಕನ್ನಡದಲ್ಲಿ ಏನು ಹೇಳ್ತಾರೆ ಅದಕ್ಕೆ ಉಗ್ರಲ್ಲಿ ಹೋಗೋದಕ್ಕೆ ಕೊಡ್ಲಿ ತಗೊಂಡ್ರು ಅಂತ ಹೇಳ್ತಾರಲ್ಲ. So yeah, it's basic. We have a Kannada in one proverb that means something can be taken by your nail like this one while you bring a big hammer to take that one. So that will happen. You are welcome to do that. You can use a UHT treatment equipment to do the hydrolysis. Acid is a corrosive one. It will corrode your system and you have to clean it, and no, we just want to have things in a very simple way. And we are not doing hydrolysis in bulk, please note that. We are talking about 1 ml, 2 ml, 3 ml. We are not talking about hydrolysis in 100 liters, 200 liters. No, nobody does it in analytical chemistry. However, if you want to do a partial hydrolysis or maybe peptide, something like that, you have to do that one. But I would not use, but UHT treatment is again different. You have a steam injection and you have a plate exchange, they are different. It's very difficult to adapt that technology to here, in my opinion. Make sense? It's a good question, but it's an overkill, that's the right word. Okay? Any question about microwave? All right, so then coming back to Asta's question here, can we use enzymes to do that one? Yes, we can use enzymes. Acids are very corrosive. We are talking about 110 degrees Celsius. We still have a problem. We degraded cysteine completely. We converted asparagine into aspartic acid, glutamine into glutamic acid. There's a lot of problems. To avoid these problems, we can use enzymes to do the job. So we have set up a method. We don't say we have developed a method. Based on the literature, we have set up a method where we use 4 different enzymes to completely hydrolyze a protein into individual amino acids. We use pepsin, we use pronase E, we have leucine aminopeptidase and prolidase. Pepsin is an enzyme, it's also present in your stomach. What it will do? It will convert this large protein into small polypeptides, a very big one into individual peptides. That's the job of pepsin. It will not convert protein into free amino acids, please note. Pepsin cannot do that job, okay. It will just convert bigger into smaller pieces. Then we add a very important enzyme called pronase E. Pronase E has both endo and exopeptidase activity. It will just cleave proteins everywhere, okay. It will just cleave proteins everywhere and convert protein into free amino acids. But some instances we also add 2 more enzymes, leucine aminopeptidase and prolidase. leucine aminopeptidase will attack leucine containing peptides and prolidase attacks proline and hydroxyproline containing peptides. These are the secondary amino acids. So, you can have a complete hydrolysis converting protein into amino acids by using this 4 enzyme combination. You can ask me this question, how did you get this one? It is all about optimization. Try so many conditions, we know this works.
Means all the enzymes at the same time?
No, I will show you that one now.
Okay.
Okay, next slide.
These steps are carried out in sequence.
Sequentially,
okay.
yeah. So, how do we do the enzymatic hydrolysis? I mean, we have set up this method, but originally this method was developed by Thomas Henle from TU Dresden in Germany, copy from his paper, but we have optimized it for our own purpose. So, we take proteins equivalent to 3 milligrams, any samples you can use, okay, make it fine powder first, and then we add HCl. Okay. HCl is basically, it creates acidic environment because pepsin loves HCl, pepsin only active in HCl condition, right. So, then we add the first day, the day 1 we add pepsin, okay, first day on the day 1 we add pepsin, on the second, then we incubate at 37 degrees Celsius continuously for 24 hours, and on the second day we add Pronace, answering your question, second day we add Pronace. Now, what happened here, first day the big protein into, you get a small peptides. On the second day we add Pronace, and that day what is happening, the small protein is becoming peptides and some will become free amino acids. On the third day we leucine, then basically will get every free amino acid, and that will be analyzed. I will talk later about here. So, the student has for 3
experiment, that is why it requires temperature and as per active
enzymes. So, all has, but we have optimized everything, we know that very nicely.
Operation is,
yeah, operator is needed, that is the one thing. But the one other thing is that you can run like hundreds of samples together, usually time is not a big problem here. It's as long as the PhD student can pipette it so quickly, then it's okay. You can run, see I have a picture here. We are running 200 samples together.
So this is the picture from your lab?
Yeah, it's a picture from our lab. So this is what we use in our lab today, enzymatic hydrolysis. Is it clear? But I know it's huh? No, the sample is basically we can optimize. We don't work with the volume. We want milligram of, and we activity of enzyme. Final volume of become 0.7 because we had the buffer and our everything here. Okay. What's that? That's an incubator. That's an incubator. 37 degrees Celsius. Yes, exactly. Okay. And it's all done in an append of tube basically. It's not this tube. I did not get a nice picture. We do it in an append of tubes. PCR tubes. PCR tubes are smaller, ml or 1.5 ml. What's that? Oh, you call it as microcentrifuge. We call it as append. Maybe append of is a we use, so append of tube. You can also, yeah, it's centrifuge tube. That's the right word, maybe.
Sir, multiple enzymes.
Yeah.
Is there a peak related to these enzyme proteins?
Yes, very good. We are also evaluating that one. You become very happy. Usually, the amount of enzyme so small compared to the amount of protein present. The amount of protein milligram, the amount we add in the micro, so it's a negligible percentage. But the biggest challenge, even if you add a amount of and clever behavior, that this is protein, right? And you add an enzyme, protein, acid. Enzyme we are using proteases, right? The beauty of proteases is that this is a protein and it is a protease. It will all the peptide bonds amino acid. Now it is becoming jobless. No substrate is present, it becomes jobless. When it is jobless, what it will do, you know? It's so, there is another here, eating themselves, autolysis, enzymes themselves, their partners, and that can also measure that one, but it's very negligible. I'll give you the data in the next slide. That's a very good. So from the enzyme, the contribution to proteins. So we compared acid versus enzymatic. No, this is a result. In case of BSA, we can see that we are actually getting a theoretical value. That means we know in BSA how many lysine is present, for example, because we know the amino acid sequence of BSA is bovine serum, pure protein. So data clearly actually producing a very high recovery. It's a fantastic technique to get almost all amino acids. We also proved it with the beta lactoglobulin. It's another milk enzyme. We can produce in ultra pure type, so it will help us to calculate theoretical or experimental. You can see they are really good. Methionine is also fantastic. We are talking about recovery of methionine. Earlier in acid, we are getting 60%, 70% and 0%. In enzymes, methionine recovery can be between 80 to 100%, provided you use the right sense, and that's the reason it takes us hydrolysis. Okay. Coming back to how contribute enzymes, contribution from enzyme is very negligible, and that we don't need this one. Enzyme stock solutions are also stable. That means you can store the, you mentioned about enzymes are expensive. However, we can make a stock solution and you can store it for a long period, and whenever you want, take a small aliquot and you do it. That's why we save money here. It's not that you take big and empty everything and it's all gone now. We can balance it in a way that it makes less expensive. But we also made a call for that, just to let you know. We have published a very nice article 2 months ago with all this data. We found that if you are running, I think around 300 plus samples with the enzymes, the cost becomes equivalent to acid. Not that expensive when you have more samples. When you have 1 or 2 samples, it becomes. All right. So important thing I have to mention in the enzymatic hydrolysis is that we still use condition. Can you see? We use the prolidase and everything like to hydrolyze. You can have some unwanted chemical reaction because you will keep the sample at 37 degrees Celsius. Protein oxidation takes place because you generate hydroxyl radical. If there is a small metal ion present in the sample, this hydroxyl radical usually attack methionine and cysteine and other amino acids very rapidly and can oxidize samples. But to do this one, we need oxygen as a catalyst. I mean, as one of the reactant. So we set up another method nowadays. So we do whole experiment in the glove box. You know glove box? Glove box is a machine and a box. You know fume hood, right? Or fume hood, you know, right? It's a box where you work so that you don't breathe the chemicals, right? You just put your hands inside. But there is still oxygen in the fume hood. Do you agree? But this is like a fume hood, but there is no oxygen inside. It's completely evacuated, and you put hand in the rubber gloves like this one. Can you see these things here? Put hand in, and you bring everything there. Completely oxygen-free atmosphere, and that will help us to minimize the oxidation because oxygen is a problem. So we have been using this one to completely remove oxygen so that you can get the highest recovery of all the amino acids. Now you're thinking about why we are doing all these things. You can just hydrolyze and get the data. No, that's not our goal in our laboratory. We want to get as accurate data as possible, as clean hydrolysis as possible to get the best result out of proteins. Okay. I just wanted to tell you that we recently published, since you guys are more interested in hydrolysis, really recommend go and read this paper. We have compared all the different hydrolysis method available in the world today, the most important ones, and we could conclude that microwave hydrolysis and enzymatic hydrolysis are the best one today compared to any other hydrolysis method. We convert to yet analyzed amino acid, which what I prepared sample preparation, we have not analyzed yet. Now I have a lot of amino acid 1, amino acid 2, lysine, glutamic acid, everything is there in my tube, and that tube is called hydrolysate. Now my sample is ready for the analysis. To analyze the individual amino acids, today we use two techniques. One is gas chromatography, liquid chromatography, and I like very briefly. Liquid chromatography I have explained in detail in last class. Reverse phase chromatography, C18 chromatography, we use the same thing, and we also use gas chromatography. In GC we use this is the GC from our own laboratory. GC gas chromatography, I have a mechanism, simply it's in this the stationary gas liquid, and the mobile phase is gas, okay, that is the only difference. So GC works based on the principle that it analyzes the volatile molecules, right. It only analyzes the volatile molecules because it has to have in the gaseous phase. But amino acids are not volatile, okay. Amino acids are not volatile. GC can only analyze volatile molecule. So we make amino acids volatile, okay. Amino acids are not volatile. GC only likes volatiles, okay. So we need to convert amino acid into volatile. To make that we do derivatization. Derivatization is nothing but reaction of an amino acid with a derivatizing agent, and that gives you a volatile derivative, okay. We do it again manually in the sample preparation. You take the amino acid mixture, add a derivatizing agent, heat it, stir it. I do not explain all this procedure in detail. You just convert amino acid into volatile derivative. The procedure is I think I skipped it, and then you get the volatile amino acid, and that will be injected to GC. Now I think back to your question, the derivatization procedure here is manual. You have to do it manually. Hydrolysis is also manually, but this is automated. You do all the injections automatically. You have an auto sample, it will inject. Exactly, you need to spend 2 days for sure, no problem. That is there.
And sample preparation is the crucial step for the
absolutely. Sample preparation is the crucial step for the chromatographic analysis. The careful, in the better way you do, you get the best result in the world. If you, everything is, if you have best in the world, preparation crash, you are done, you get a wrong result. So sample preparation, and that is the reason why I have been explaining about hydrolysis condition. So much time we spend on that one, because if you mistake that, something is wrong. Fastest chromatograph of my life, I got it during my PhD. So you can see that how beautifully we have separated individual amino acid. It is a method from Phenomenex actually, they have the column and other things, but you can see that we have separated 26 amino acids. 20 normal amino acid and 6 non-proteogenic amino acids we have separated. No, this is something I just labeled just to highlight aspartic acid and glutamic acid. It's because aspartic acid and glutamic acid are giving umami taste. You know that umami taste, the fixed basic taste, and that is because of aspartic acid and glutamic acid. We were at that time we were measuring that term. So that is the reason I highlighted. Is it clear? So this is how the chromatogram look like. So you can see individually alanine, valine, proline, glutamine, ornithine, lysine, histidine, cysteine, amino adipic acid, hydroxyproline, asparagine, everything is separated one by one, and the area under the peak is directly proportional to concentration. By using a calibration standard, you can measure the amount of essential, non-essential and conditional essential amino acids. Is it clear about what is we have done? We have hydrolyzed and we ran the GC. Before running the GC, we need to make them volatile and also make a little bit non-polar. For that, we do add a derivatizing agent, and that takes a lot of time. I was doing that when I was a PhD student. For one sample to derivatize me, you take a sample, add a buffer, add a solvent, shake it, vortex it, and take the supernatant out, again centrifuge it, again cap it. For one sample, it might take me around 20 to 25 days. As a 100 with samples, so this is a number. Maybe industries analyze 1 or 2 samples, so they dedicated people for that. As a student, 100 samples for normal number for PhD lab. It is time consuming. I from morning to late in the evening derivatizing, and that's too much consuming. Then we have set up another method that will do most automatically. That is the HPLC method. In this case, we derive the OPA. OPA is orthophthalaldehyde agent. It's very fluid. Now we have an amino and we cut and then new product. Yes, the OPA can be used in a many, just strong chemical. What's that? Yes, it just attacks the free amino groups. The free amino assay can be done with OPA. Same principle. It reacts with the free amino group of a protein, or in this case amino acid, and becomes this compound. Okay? And this compound is fluorescent active and also UV active, and that you can easily detect by chromatography. Okay? For secondary amino acids, for example, for proline and hydroxyproline, OPA does not react with them. It only reacts with primary amino acid. For that, we use another reagent called FMOC. It's a chlorinated compound. You can see that one, and that selectively reacts with the secondary amino acids after OPA reacts with the primary amino acids, and we get another compound, and that gives the peak for hydroxyproline and proline. We did derivatization for the into volatile, making it less polar. Derivatize, convert them to fluorescence active molecule, because are not absorbing and tyrosine and UV light. Difficult to detect them with HPL. So detect will be OPA, and it also clarity. It helps us to separate in the reverse phase chromatogram. And see the chromatogram of the HPLC amino acid analysis. Can you see? It's also so nice. We could chromatographically separate every single amino acid here, except we don't have cysteine here. That's it. The beauty of chromatographic analysis with the HPLC here is that we use an autosampler here. Can you see? For derivatization, we need OPA, we need FMOC, we need borate buffer, and we need acetic acid, and we need sample. Okay? These are the 5 different things we need to do this chemical reaction. Okay? We need OPA, we need FMOC, we need a buffer, we need acetic acid, and we need sample. Manually, how will we do? We take the sample 100 microliter, add OPA, stir it, wait for 10 seconds, add FMOC chloride, stir it, wait for 10 seconds, and then add, before that you have to add a buffer. To stop the reaction, we will add acetic acid, wait for 10 seconds. 200 samples will take me an hour. And I will do always, right, every does after samples. So this is nice and automatic. And you just take from here, microliter, mix it in the needle, and take microliter. It will mix in the. You say it's all scripted. It will do everything automatically and saves my life a lot. We are talking about running thousands of samples nowadays in our instruments. We have 2 dedicated instruments to do the amino acid analysis. when you say thousands of samples, you know how much time we save here, especially in Denmark, time is money. Salary is very high because of the time is, if you want to hire a person to do the derivatization, to high salary, but automated is very cheap. Makes sense? Okay. We will take a break in 5 minutes and then we will get back. We will take a break in 5 minutes. Just to conclude, there are a lot of amino acid analysis methods. Mentioned the analyzer, your amino acid analyzer. It is nothing but an instrument similar to HPLC. They have usually the samples. Get a chromogram automatic. It's a different machine, that's it. And derivatizing agent there they use ninhydrin commonly and we use OPA. That's a small difference. So rest everything is the same. You still do a manual hydrolysis.
Also searching for sample analysis of my student work, and the charges for amino acid analyzer are very high.
Yeah, the India. Equipment.
What if that if at our lab give it to lab whereas GCMS is there and with FID detector.
Yeah.
So can we get it analyzed?
Yes, in principle these derivative methods are not stable. So it's always recommended that you do derivatization on the same day or at least maximum previous night and put it in the freezer.
Okay.
So you can't ship it to some other place. If the shipping takes you 2-3 days at room temperature, you are done. But we do use an internal standard to avoid all these problems, but still I don't recommend that.
Okay.
So derivatization has to be better done in the same lab to avoid the degradation of strategy. You go to that derivatization there, immediately give it to them, they can analyze. I would advise that.
Okay.
So just to conclude my analysis here, there are a lot, but the biggest body is hydrolysis, because if you use a right hydrolysis, you will give a data about acid. Because there may be methionine is present in your sample. Because of sample preparation, you just degraded everything and you tell to people that my sample has no methionine. Or you do a mistake chromatography, nice methionine peak, done, say my sample has methionine, and you are misleading. So you need a very good analytical experiment, the right and it is time. It has its own problems, its own advantage and disadvantage, but today we use the same thing. I hope somebody will make completely automated in the future and 20 years. Opinion. So with that we will break, come back after exactly 14 minutes, shall we say 15 minutes, okay? 25 we will be back. 11:25 minutes we will be back and we will resume the lecture. So to go for restroom and or something, just go, okay? You don't have to ask my permission, don't hesitate, just go and come back, just to let you know, okay? Because I'm taking long classes and I don't expect you to sit, and you are welcome to go back and come back, okay? Of course, you will be on the video, but just be aware of that. Maybe your teacher is watching whether this guy is listening or not. Okay, we will back and we will start in lecture. Continue with about nutritional evaluation, acid profile and bioavailability. Heard about nutritional evaluation. So protein is depends amino acid composition, and the second one is digestibility or bioavailability. We just heard about essential amino acid. There are 9 essential amino acid, and we discussed about there are a lot of different techniques are available today to analyze essential amino acid composition, and that contains 2 technical aspects. The first one is hydrolysis, and second one is amino acid analysis, that is usually done with chromatography. This is what we heard about in the first half of today's session. Any doubt about it? Any questions? Is clear, right? Now we will move on to the second part of the protein quality evaluation, that is digestibility. I will try to take you a little bit quickly here. Digestibility evaluation can be done in 2 approaches. One using animals, second one is without using animals. If you use the digestibility analysis, if you use animals, if you use in vivo experiments, right, that gives you one set of data. So, animal based experiment is often called as in vivo or in vivo, right, you have heard about it. In vitro is means in the laboratory without using animals, human beings and so on. Vitro means glass, right, in vitro means in glass, means in laboratory. So, digestibility evaluation can be done both by using animals or just using glasswares in general. Okay. Animals could also be human beings. Humans can also be used to evaluate the digestibility, but we often do not use them. Why? Because humans are more expensive and it is very difficult to train them. I will get back to you on that later. Okay. So, we often use animal models, especially rat and piglets. A rat is a rat, you have seen, right, and they are crazy, they are nice. Some of the digestion that happens in the rat gut to a certain extent can be translated to what happens in our body. At the end of the digestibility experiment, the end goal is to understand what exactly happens in human body, right. We do not care about what exactly happens in rat body, but we can't use humans in some cases, that is why you use rat. But please note that if you use rat models, it is not necessary that the knowledge you get is completely 100% translatable to human being, but we still get some sets of idea. But apparently pigs are more equivalent to human beings. That is a bad statement to make, but at least their digestibility apparently to a certain extent can be translated to the what happens in our body. So, we can use pigs as a model organism. Human beings can be used, as I said, they are expensive, difficult to train. For example, I would like to evaluate digestibility of meat analogs, and I will recruit these 2 gentlemen, okay, and I will give them meat analog, but silently in the night they would eat something else, right, I will have a problem, right, and somebody, the director was telling that somebody will go to forest and do something else there, right. And these are all the problems, right, we have. So, I do not say we can't do a humor intervention today, we do that one, but it's super expensive and super difficult to control human beings compared to pigs and let us say rats, but we still do that experiment with human beings. And then the most cheapest one is in vitro method, nothing is involved, human beings are involved just to do the laboratory preparation, that we do not eat anything there. But still you can get a certain information from the in vitro experiments. Now, I will go through different techniques we use today to evaluate the digestibility of alternative proteins as quickly as possible. If there is a question, raise your hand. Okay. Let me begin with the methods approved by FAO and WHO back in the days. They approved a method called PDCAS. Have you heard about the PDCAS? Protein digestibility corrected amino acid score. It is a method to evaluate the digestibility of a given protein, and back in the time 1989, I think it came in 1990 if it was approved, if I am not wrong, it came into existence. They thought this is a fantastic method to evaluate the digestibility of different proteins from different matrices. Okay. The idea is simple, it is actually a mathematical score based on the mathematical data and also biological data. PDCAS is nothing but amino acid score multiplied by fecal true protein digestibility. Okay? Amino acid score multiplied by fecal true protein digestibility, and you will ask, it has two components, amino acid score and true protein digestibility, and they are determined by two different methods. Okay? Amino acid score is nothing but it is the milligram of limiting amino acid, that is the essential amino acid, milligram of essential amino acid in 1 gram of test protein, that protein you want to evaluate the digestibility, divided by the milligram of same amino acids in a reference protein, and that value can be already FA has given. If you want to have let us say threonine, it should be 34 milligram per gram, that is with the denominator. Am I clear? So, amino acid score will be calculated initially, that is nothing but milligram of essential amino acid, the given one, per gram of sample. How do we calculate this essential amino acid? Same as previously, amino acid analysis. You do the amino acid analysis of your protein, calculate the essential amino acid content and use it in this formula, right. The second part of this procedure contains fecal true digestibility, right, and that will be done using animal models, especially our friend rat here. Okay. They feed the protein to the rat, okay, and the rat will digest the protein, right. Some portion will be absorbed and then it will get the fecal amount that will excrete something. Okay. The amount of nitrogen, because nitrogen is coming from protein according to them, is directly proportional to the digestibility, I mean inversely proportional to the digestibility in this case. If more and more protein is digested, more and more is absorbed, and the less and less will come in the poop. Do you agree? That is the whole idea about evaluation, and those two factors are multiplied to get the PDCAS. Am I clear? So, yeah, any question? So, PDCAS is nothing but it is a combination of amino acid score multiplied by the true fecal digestibility. They are using fecal digestibility here, means they measure the nitrogen content in the feces, right. If you have a less nitrogen in the feces, means in other words it is more absorbed, more digested, and that will give us an understanding about PDCAS. But one important thing we have to observe here, I will come back to it in a later point, they are not measuring the amino acid here in the feces, they are measuring the total nitrogen. Keep that in mind. They are not measuring the individual amino acid, they are measuring the total nitrogen. Keep that in mind. The second thing is that they are not measuring anything in the gut, what is happening exactly, they are measuring in the poop, in the feces. Am I clear? There is little bit dodgy thing here happening, and that is also a disadvantage. I will come back to that later. But for now, is it clear how do we measure PDCAS? Just to give one, this is the same thing, it is just for your information. Just you can see that the PDCAS numbers of different, they are actually truncate the final value to 100% or 1. If you have a 1 or 100%, it means that it has the highest digestibility according to PDCAS calculation. That is the maximum number you can have. You can compare here is that casein has 100% digestibility, soy has 92%, wheat gluten has a very poor digestibility, 24%. So, this is the PDCAS value. Am I clear? This I have already showed you in my previous lessons. So, PDCAS can be used to compare digestibility of different alternative proteins, but today PDCAS is not used. You still see a lot of data on PDCAS, lot of papers still show the comparison PDCAS, but FAO and WHO now agree that PDCAS is not the best method in the world to measure the digestibility. Two reasons. Number one, how the human digestion happens. You eat here, something is digested in the stomach and intestine, from the intestine it goes to the blood, okay. And in the PDCAS what they are measuring, okay, digested something is went to the blood in the rat model, and then they get it in the poop. What they are measuring here, it is the nitrogen, but nitrogen can come also from different sources in the body, and there is a lot of gut bacteria. Actually the absorption happens in the small intestine. Now, if something is not absorbed, it comes to the large intestine, and the large intestine there is a lot of things happens. Their things can also be digested, but they are not bioavailable, right. And then what you measure the nitrogen here might be a wrong one. So, usually the PD-Cas value could be overestimated, because it shows that, oh, there is no nitrogen left, oh, there is a lot of nitrogen left, and that might give you an indirect wrong answer, because the nitrogen can also come from different sources, not necessarily because of the digestion happens in the gut, in the small intestine or in the stomach. Am I clear? So, they thought it is not a good method to measure digestibility, and recently in 2011 they come up with a new method called DIAAS, is called DIAS. Okay. DIAAS has tried to avoid the disadvantages of PD-Cas. The full form is digestible indispensable amino acid score. Indispensable is nothing but essential amino acid. Digestible essential amino acid score is called DIAAS. Now we are using in 2000. It has its own disadvantage, but now we strongly believe that we should go with this method compared to PD-Cas. Am I clear? What is the difference here? DIAAS measures ileal digestion. Ileal is basically you have a small intestine, the end of the small intestine is called ileum, the last part, that is a transition between small intestine to large intestine. They measure, they take the sample in the end of the small intestine to measure the digestibility, that is the main difference. They do not use the feces to measure the digestibility, they use the ileum digester to measure the digestibility. The rest is somewhat similar, you know, look into the formula, okay. And the reference protein value has also changed. I hope I have the data is here, okay. The most important thing here is that they recommend the DIAAS has to be measured with pigs or human beings, in the worst case only rats. They recommend, it is nice to use it human beings, but human beings are very expensive, but most of the literature I found they use pigs. Pigs are little bit less expensive compared to humans, easy to maintain compared to humans, but please note that in vivo experiments are damn expensive. All in vivo experiments are super expensive, is simply because you need a dedicated laboratory to maintain animals, right, that is the one expensive thing. The second thing is that you need to get also ethical approval. I forgot to mention that. You can't simply feed pigs and I will measure digestibility. No, government will take you to jail, right. You can't use rats, you can kill rats at home, but not in the lab, right. That's another story, right. You can eat pig in another way, but not in the labs. Don't eat pigs in the lab, don't slaughter there. You have to take an ethical approval. Am I clear? So, that is the reason it becomes super complicated. The ethical committee will not give you an approval unless you justify why you are using these many pigs, right. Yeah, any question? Yeah.
Sir, how it is possible to get this ileum in practical?
How it is possible to?
How it is possible to get this portion after the small intestine?
Exactly. How do we get this portion out? Because we are measuring the digestion in the ileum, right, the last portion of the intestine, what we call, I think it is also called duodenum or something. Duodenum? Duodenum, sorry, I can't pronounce that word since my childhood, forget about it. Okay, that one, okay. How do we collect that is basically in the pig, they use this something called cannula barrel. Can you see here, I have written it. Basically they do not kill the animal to collect the and then do the dissection and collect the, no, they do not do that one. You can do it in a more easy way. They connect a stainless steel or plastic cannula barrel to the right region of the pig and they collect the digester every half an hour. So, you can do it. Pigs are little bit unhappy, but they are not dying at least. Am I clear? That is how we collect the digester using, I hope I answered your question with this figure, the same figure. Am I clear? So, the difference between PD-Cas and DIAAS is DIAAS measures the ileal digestion, not the fecal digestion, plus they measure individual amino acid in the ileal digester. They are not measuring the nitrogen, they are not measuring the total nitrogen, they are measuring the individual amino acid, how much essential amino acid is present in the digester. That gives us a better information about the digestibility. So, nowadays the recommended method is DIAAS. Am I clear? But unfortunately, I could not find a lot of data on DIAS because it is very expensive experiment to do. We do not have much data on alternative protein world, DIAS value. You will find a lot of data on PDCAS. DIAS is very limited data is available, but I found one research article, they were trying to compare meat versus meat analog. Okay. They fed this poor pig both with lean meat and meat analogs, burger analogs continuously for 9 days, and then they collected the digest on the 9th day if I am not wrong, every half an hour, and then they did the amino acid analysis, and then they measured the DIAS value. You can see here is that the lean beef, this is the digestibility about 90%, and 93% lean beef is, I think it's a moisture protein content, right, 93% lean beef, I think it's a protein content. It's have extremely high digestibility. And the burger, these are the most commonly sold meat analogs in the US market, Impossible Burger and Beyond Burger. They are not super bad, you can see, compared to meat. They are still having a very good acceptable digestibility. Am I clear? So, meat analogs are not essentially bad in terms of digestibility. The protein content in them was varying. Any question here? DIAS? So, in the case in the future, for example, if you want to extract proteins from insect, microalgae and something, you want to do the digestibility, recommended is DIAS, do not go for PDCAS. There is a nice table, I found it again from this research article, they were comparing DIAS versus PDCAS. You can see that they are very comparable, but there are differences. Okay. One important observation you can make here is that DIAS value can go beyond 100%. That means that the value can be higher than the reference protein, but PDCAS they are always in mathematically they convert into 100%. So, it will never go beyond 100%. One is 100%, please note that one is 100% here. So, this is equivalent to 1.14 or 114%. So, this is the difference between PDCAS and DIAS. They are similar but dissimilar. For you can see that chickpeas, there is a big difference between PDCAS and DIAS. So, we should be very careful with which method you are using to evaluate the digestibility. Am I clear? Alright, these are the two in vivo methods, and this is a nice article that shows because we are using rats or pigs, how much is equivalent to human beings. They made an experiment here to compare the digestibility in pigs versus human being, and they found a nice correlation. Even they have used lot of different proteins, you can see that the correlation is really good. So, that indicates that you can use pigs as a model organism to evaluate digestibility in human beings. Human beings are crazy, pigs are less crazy, however, still pigs has to be given this blah blah is not good. Human beings also they take digester in a different way, they insert something and they can take it. I do not want to be in that experiment anyways. So, make sense? So, you still can use pigs. Now, these in vivo models are so expensive, it takes a lot of time, lot of approvals, it is not easy to do. Not every lab can afford in vivo models, maybe 1 in 100 labs can afford in vivo model, I am telling you that one. It is easy to tell, but difficult to do in vivo things. So, but we are working a lot nowadays, lot of alternatives, we can't wait for in vivo. In vivo for one protein takes you 10 days, 100 days of approval and lot of other things, it will take you forever, but we are extracting proteins every day nowadays. We need another method, right, another easy, convenient, cheap, but still acceptable method, and that is called in vitro method, that is done in the lab. Hundreds of in vitro methods are available, but today we are using InfoGest protocol. It is an harmonized protocol published back in 2019, I think. They published the first paper in 2014 with a nice protocol saying, okay, you can follow this particular method. And 2019 they published in Nature protocol a second protocol with an advanced information that we call InfoGest protocol. Most of the laboratories today they use this method just to show that everybody follows the same method, that is it. For example, in vitro is nothing but basically in the body, this is our This is not snake, it's our body, huh? Okay, this is the body, right? We eat here and there is a stomach here, you get a pepsin and with a lot of enzymes in the small intestine, trypsin, chymotrypsin and everything, right? We just want to replicate the same thing in a glass. Okay, this exactly happens in the human being. We want to do it in the glass, in a test tube. Okay, we use the same enzymes, pepsin, chymotrypsin and everything, but everybody is doing it in a different ways. She is using pepsin coming from somewhere else, and she is using chymotrypsin at very high activity, and that person is using a different temperature, and this person is using a different buffer. Then we can't compare the experiment from him with her. She is doing a different thing in her laboratory with the same chickpea, and he is doing different thing in the same chickpea in a different way. Then the digestibility will become different, right? She says it's 80% digestible and he says it's only 40% digestible. Whom do I believe? To avoid this problem, we want to have an homogeneous protocol, and then they come up with this idea called InfoGest, and most of the people are using now this InfoGest. You will see it in the internet, right? The protocol is not simple, but it's a very nicely written protocol. They use oral phase digestion. They use first they prepare the sample, they use the oral for digestion, they use salivary amylase here. Then they do the gastric fight digestion in the stomach, they use pepsin, they use calcium in a coenzyme, and they use pH around 2 something like that, okay 3 here, can you see? They maintain the pH. And then they come to the intestinal phase, they use chyme and they use bile acids, they use trypsin, they also use other pancreatic enzyme like chymotrypsin. Am I clear? But everything is done in a test tube or a falcon tube. They do shake, they do mix everything. I don't teach all these things, but it's just replicating what happens in the body in the falcon tube. There was one more slide I deleted unfortunately to answer this question. It takes a couple of days because the sample preparation, the first this one, it will take you 1 full day. There is a lot of buffers, acids, everything you will do, and this will take another couple of days. There is actually, if you read this protocol, they were clearly written how many days it will take. It is not done in half an hour, okay. I am making the things more easier for you. Yeah, yeah. Do they, oh, it is written here, sorry. It takes a, yeah, exactly. Yeah, you can do it in 1 day, but actually the sample prep takes a lot of time basically. 1 day is basically to stir it, to mix it and everything. I have done it myself, not exactly this protocol, in other modified protocol before. It takes a lot of time. It's because the yeah. Yeah, that is basically, it's clearly, I think, yeah, 5 hours exactly, something like that one. But the problem is that what I found, the reality, from gastric phase to intestinal phase, you need to change the pH from 3.0 to, let's say, how much they say? 7.0. That pH adjustment is a crazy thing, especially when you have a lot of proteins, because proteins have a buffering capacity. You need to add drop by drop sodium hydroxide and suddenly it will increase and suddenly, and that takes me forever. But nowadays they do it more automatically with the automatic titrators are available that adjust automatically, dynamically it will adjust. But if you want to do it manually, it's easy to say, difficult to do, but people use it. Yeah. I think so too, but we use it in a 50 ml falcon tube in our laboratory, a small bioreactor of falcon tube, centrifuge tubes, we use that one. And it's very difficult to handle too many samples, I also found that, because you can't simultaneously adjust the pH of 10 different samples, because time is also running out, right. You can't incubate it in more pepsin now, because you are adjusting the pH in one tube, pepsin is acting in other tube continuously, because you need to bring pH from 3 to 7 quickly, but you are adjusting half an hour with one tube, other tube is decreasing. Yeah, that's the reason. This is the biggest challenge with this method. Unless you have an automated machine, all my students are not doing it this one, but I had some master student doing it before. They were not happy because they can't handle too many samples. That's a disadvantage, but this is a problem. That's okay. But still, it saves. Compared to pig experiment, it's fine. The pH adjustment is a nightmare, at least for me. When I did it, was a big, I hated it actually. Adjusting the pH is a nightmare here. But apparently there are some automated stuff nowadays to adjust the pH. They will do it, that's called titrators, they are available now. But still it's a recommended method, it's really nice, you should follow this method. It's the same protocol I was telling. And you can see here is the digestibility of different samples here. You can see that this is a milk protein concentrate, has a in vitro digestibility of around 90% or 85% here. And the lowest is here, HC. What is HC? Can somebody read HC here? House cricket is an insect. Okay, insect has a lower digestibility according to IVPD of that particular insect, right? So we can compare how different samples have a different digestibility. Please note that I am not a big fan of in vitro digestibility to find out the true digestibility. It never gives an information about true digestibility simply because it never considers how much is absorbed in the body, right? PDCAS and DIAAS considers how much is absorbed in the body, it only considers how much is broken down. However, it is a nice experiment because it has a good correlation with the PDCAS or DIAAS, I will come back to that one. However, it is very nice experiment to do to compare different samples. For example, in your own laboratory, you want to compare ultrasound assisted extraction protein with salt assisted extraction protein. What is the digestibility difference? You want to compare because you are working with the pea, soy, chickpea, insect, microalgae. You just want to compare how different they are. This is a fantastic method because you can run them parallelly in the same department with the same person, you get the same data. So it is nice to do the comparison. I really like this method, but another disadvantage here is that if you have a small difference in the digestibility, it cannot measure properly. For example, if you have two proteins, 90% digestion and 95% digestion, then the standard deviation is always 5–10%. So if there is a small difference in the digestibility, I am not a big fan of InfoGest. So it cannot show, this is our experience, maybe other people have a different experience, I do not know. For our experience, if there is a small difference, it cannot, it is not very sensitive method to see the difference between protein to protein. Any question? But it can be used for comparing different proteins. I can skip this slide. Just to show that there are lot of, InfoGest is a static method, means everything is static. Nothing is moving from stomach because there somebody in stomach actually there is actually pushing and pulling things happen in intestine, and those things we are not mimicking actually. But now there is a lot of dynamic models are also available. Dynamic models means it's trying to mimic in the machine like in your stomach, moving things like this, everything can be done. So a lot of machines are also available to do it more dynamic model today. I do not explain that in here, but they are all complicated. You can see that I found it in the internet. See, it looks like a stomach, can you see? And here is also looking like a stomach, and maybe this is intestine, I have no clue. This machine I saw it in China in one of my collaborators' laboratory. They have a dynamic model, it mimics the digestibility. They have a mouth and a plastic stomach and a plastic intestine. They adjust the pH inside. Many things are being done just to mimic because they can't buy human beings, expensive, crazy, huh? All right. This was the paper. They are trying to compare whether InfoGest is used to complement DIAAS. DIAAS is very expensive, time consuming method. Can we make DIAAS as an in vitro method? They were trying to make this experiment. They were kind of successful, I would say, because they could show here is that in vivo method and in vitro method had a very good correlation for most of the sample, but not for everything. Okay. If you take let us say bran here, they could see a good correlation, right. So you can use InfoGest to complement DIAAS. You were welcome to read, I do not go into detail for this paper at the interest of time, but you can make DIAAS in vitro. It is not approved by FAO or UN, WHO or let us say other regulatory bodies, but people are trying to make DIAS in vitro. Am I clear? So, these are the things we covered today. We are talking about, just to sum up everything, we are talking about protein, important is essential amino acid, other important thing is digestible, D is for digestion. Digestion can be evaluated by in vivo and in vitro, and in vivo what do we have? Two methods, pigs and rats, let us say PD-Cas and then DIAAS. In PD-Cas is we often use rat, in DIAS we often use pigs. I think I did not read the regulatory guidelines in detail though. If I am not wrong, they suggest, it's nice to use human beings, if not pigs and if not rats, something like that, but often papers use, all the research articles use pigs to the best of my knowledge. PD-Cas has a disadvantage. What is the disadvantage? They measure the nitrogen in the feces, that is not directly it will mention the digestibility, you have a problem there. So, the FAO thinks it is not a good method now, so then they came to DIAS and today we use DIAS. Advantage is we take the ileal digester, so we measure the real true digestibility and we also measure the individual amino acids there, so it gives a realistic answer about the digestibility. The advantage of in vivo method is it gives almost real digestibility. The disadvantage is time consuming and extremely expensive. So, we have to go for in vitro methods. A lot of in vitro methods are available if you read it in the literature. The most commonly used one is static and dynamic. I did not teach you dynamic today, it is also expensive. Static is InfoGest. Okay. InfoGest is the static method we are using today, a harmonized protein. There is a lot of ring trial is also going across the world. What they are doing at the moment is that they are trying to supply samples to different laboratories across the continent and to find out, okay, everybody gets the same data. So, a lot of things are going just to make sure that we use harmonized protocol throughout the world. It is a nice initiative in my opinion. There is also dynamic model with a lot of reactors and so on, but this is a current state of the art, I would say. Most of us use InfoGest. Yeah. Mic for him please. Yeah.
Sir, how do we measure the diets of true ileal for in vitro model?
Good question. That is not the part of the in vitro model. You do not measure it, but it is basically a correlation analysis. If you go back into this paper, they do the amino acid analysis after the digestion. The individual amino acid they measure by HPLC I think, and that you try to find a correlation with the in vitro. They do not measure, there is no pig models here in the in vitro. It is just to see whether it is going hand in hand, a verification method. Yeah, exactly, mimicking thing basically, just to save money and time here. Yeah. There is no, in vitro never replaces in vivo. It is not 100% because you can see the correlation here, some things are going far away. Okay. Yeah.
Sir, that protein digestibility where the fecal matter nitrogen content we are calculating, no?
Yeah.
But if we go for same HPLC and all, how much amino acids are
we would have done that one, but that is not they recommend in the PD-Cas. At that time I would say they had a different idea because at that time everybody was measuring protein based on nitrogen content, Geldahl's method. That was the state of the art at that time. I am talking about 1989, right. They write the recommendation based on what was right at that particular time point. It is not maybe the correct one because science evolves every time, right. Maybe what they might come and prove DIAS is absolute rubbish after 100 years, I have no idea. So, then we need to use the new method at that time. So, they would have measured this one, but the problem is that as I said, when the amino acid does not absorb in the body, it goes to the gut, the large intestine. In the large intestine, we have lot of bacteria, they degrade amino acids into nitrogen. Now, we get a misleading data. Then if you measure the amino acid analysis of the fecal analysis, you might not see some amino acids, then you think they are absorbed. but they are not absorbed, they are degraded by the bacteria. So, you still give it misleading data, and that is the reason why DIAAS uses the sample before reaching the large intestine, then they do the amino acid analysis. So, measuring the amino acid at the fecal matter is not a good method according to me, because you can't avoid the problem of bacteria here. I hope I answered your question. Any other question? Looks very complicated, right? Alternative protein world. See, extraction, production, processing, analysis, digestion, bioavailability, what not we have to do. It is not the end of the story. I have one more lecture. Okay. So, we are going back to the next lecture today, that is lecture number 11, allergenicity and anti-nutritional factor. We have extracted protein, we have evaluated it, we have digestion analysis, we found my protein is 100% digestible. I am happy, but that is not the end of the story. Proteins including me, right? Some proteins bring allergenic properties. Some proteins carry anti-nutritional compounds together with them, and that can compromise the overall nutritional quality of the alternative proteins. Does that make sense? Extraction, purification, analysis, digestion, bioavailability, everything is ticked mark. That is not the end of the story. Yes, the bioavailability, okay, I have a term definition here. They use it kind of an interchangeably. Where is the definition? One second, sorry. I don't know where did I teach that. It was in the first slide. Right. I'm not getting that slide. Okay, basically, okay, there it is. Yeah, when we say bioavailability, basically the portion of that amino acid that has crossed the blood intestinal barrier and that reaches the blood, it's actually bioavailability. When you do the ileal digestion, DIAS and PDCAS, it considers the bioavailability. So, and these terms are used interchangeably, but they are similar but not the same. Does it make sense? I am not excludingly telling what is bioavailability and other things. It basically covers by the digestibility. All right. So, we are moving to the next lecture of today. I will try to cover as quickly as possible so that I will not eat your lunch break. Okay. All right. Two problems has to be solved to make alternative proteins are commercially available, human beings can consume. The first one, the problem is allergenicity, second one is anti-nutritional factor. Allergenicity, you must be knowing that, right? For example, some people say I am allergic to milk, right? I have a, maybe should not be recorded on a camera, I am allergic to one compound. I was not allergic before. If I eat apples, right, my lower lip becomes swollen and my throat starts itching. Okay. I have a friend, that person, if he or she, I should not name the person also because we are on camera, if that person eats crab or shellfish, whole skin becomes red. And there is a boiling here, right? Allergic, people are very allergic. We are talking about Maggi packet, and as I said, if you took and look into the ingredient, some things they write in the bold, be careful. Nowadays, if you go to restaurant, they clearly ask you, if you are allergic, please let us know before. They do not tell it in the small restaurants, they do not care about you, no, they do care about you, but that is another story. If you go to hi-fi restaurant, they ask you, are you allergic to some kind of ingredients? You say, oh, I am allergic to nuts, then they do not use nuts in the food, right? Because allergenicity is one of the biggest challenges that we are facing today. because sometimes they are less severe. If you have a small bubble on your lower teeth, it will go in half an hour for me. It's not a big, I don't care, to be frank. But some people get breathing problem and that can be life threatening. Am I clear? That can be life threatening. So allergenicity has to be taken care in a very careful way. In some cases it is a less problem, in some cases it is really a severe problem. Okay? There are 8 foods in America they classify as big 8 allergenic food: wheat, fish, milk, eggs, peanuts, soybean, tree nuts and crustaceans like crabs and shellfish. They are allergic, and the restaurant should ask before if there is any seafood product and something like that. They label it carefully. And I think in the Americas they also have a rule, they have to mention which one is allergenic in the packaging. I don't know if there is in India. It's also in India now? Okay. FSI has, when I was I'm an Indian, of course, it was not there at that time. They were not very strict about that, but now things are very strict. Okay. Now, what I want to tell you here is that alternative proteins can also be allergic. Can you see here soy? Where is soy? See? Soy can be, see, soybean is allergic, but alternative proteins we are getting ingredients out of it, but still we need to be careful whether they are allergic. Am I clear? Why I want to tell you, but I will come back to that one a little bit later. I do not teach you about how allergic happens. There is a lot of big figure, but when you are free, go and read all these things. But I will tell you in a very simple word, in 1 minute, how allergy happens. Okay? Forget about this big figure. Let me try to simplify it. There is something called allergens, okay, and something called body, right? We eat food as food, not as allergens. Okay, we don't know whether it's allergen or not. We just eat it as a food. When it comes to stomach, there is a digestion or bioavailable or many things will happen. There is a lot of absorption. Many thing happens in your body, right? There is something called antibody in our body, right? Antibody. Okay, and they are very specific to antigens, right? You have studied in immunology lectures, right? When this antibody sees some molecules as they are maybe a food particle, imagine protein is actually good one, but sometimes body thinks it has an antigen, it has an, okay, then they interact with each other, okay, then the immune will trigger in our body. Body thinks, oh, something bad is coming. The body thinks something is bad is coming, I need to do some response now, right? Then they start releasing histamines, right? These histamines are released continuously and that will give you so called allergic response. Now when you get allergy, they say that go and take cetirizine, antihistamines, right? It is the same phenomenon. So somehow we have to reduce the histamine content, and that is how we can create allergy. See, antihistamines are very important because especially they give it when you have a running nose and so on. If you get a bubbles after eating, they will act so fast, within half an hour or 1 hour you get less response, right? I always carry histamine in my back pocket because I am allergic to certain things. I am allergic to dust. So this is exactly the mechanism, right? There is something called antibody, they want to protect you, they are all protein, they want to protect you by some unwanted things. Sometimes they go crazy, they are drunk maybe, right? I don't know. They think food molecules also as allergen or antigen. So when they interact, they create histamines and that might be problematic. Now, plant proteins can be allergens, act as allergen, not for everybody, it's very individual thing, not for everybody, not universal, okay? And plant proteins can also carry some other things, they can also act as allergens. They can create immune response, can damage your body. Am I clear? So this is about allergenicity in 2 minutes, but your immunology professor will talk about 2 months about all these things: innate immunity, active immunity, passive immunity, immunoglobulin E, and there are many things are involved. I am not an immunology professor for now, for today, okay. So how do we evaluate the allergens? Proteins can be allergens. For example, soy proteins contains hell lot of allergens, okay. You can use STS paste to evaluate, you can use size exclusion chromatography to evaluate them, you can use mass spectrometry to evaluate them, okay. some proteins are allergens. And you can also use allergenicity evaluation by using immunological techniques. Have you heard about ELISA? Enzyme linked immunosorbent assay. It's purely based on antibody antigen interaction, right? So, your protein might act as an antigen here, and there will be an antibody specific to that antigen. Reaction takes place, color change happens, they measure antigens and antibodies. Does that make sense? And I do not explain this one. So, people often use ELISA assays and protein characterization to find out how much allergens you have in the food samples. Am I clear? Now, just to show you, I think there is one slide got deleted. I see one slide got deleted, but that is okay, I will explain you. Now, please note that you have a soybean, okay, and soybean has proteins inside, I told you last time, right, and proteins are located inside soybean, right. Allergens are also located inside soybean, and we do not eat soybean directly, do we? You get really a problem. It is cooked. So, allergens can be inactivated, I will talk about that later. But how the plant based world is acting today, how they are producing, they do not use soybean directly, they get soy protein isolate, right. They get soy protein concentrate. How do we get that one? Go back to my second lecture. We do dry fractionation and we do wet extraction, right. During extraction what happens, let us say dry fractionation, you have soybean flour, you get fine powder and coarse powder. Now, the anti-nutrients or allergens are actually fractionating, right, and the same thing happened with the wet extraction. There might be a possibility that you are extracting or separating, you are also concentrating unwanted things in each fractions. So, keep that in mind. It is not that when you do purification everything is gone, you may be up concentrating unwanted things also. Okay, I will explain that in detail when it comes to anti-nutritional factor. Just to let you know that you may reduce the content of allergens, but it is not that you are completely removing them even after doing the extraction and purification. So, how do we remove the anti-nutrient? I am sorry, I am mixing up allergens and anti-nutrients. I am very sorry about it because I am also hungry now. So, allergens, how do we inactivate them? A lot of methods. The easiest one is thermal denaturation. For example, you heat, proteins will be denatured and then they might not interacting with the antibodies. Okay. Lot of methods are being done. You can do the polyphenol cross linking. For example, the antibody has a specific binding sites. Now, imagine that is binding to this amino acids. Now, you convert this amino acid into something else. Now, the antibodies cannot interact with them. So, you can reduce the allergenicity. I do not explain all these things, it is out of the scope for me today. Just to let you know that there are a lot of different strategies to avoid allergenicity. So, food has to be processed. I was talking about a dilemma between processing and non-processing. If you do not process the food, you have problem with allergenicity. You can't eat everything raw. You can't eat everything gently processed. Sometimes we need to do heavy processing to kill some unwanted things. For example, allergens. Am I clear? You can also use enzyme hydrolysis because this is an allergen protein. Imagine you do the enzymatic hydrolysis, you get small peptides. Maybe they are not interacting with the antibodies. So, you can reduce the allergenicity. Am I clear? Yes, there is a question.
So, if a person is allergic to protein,
yeah,
can he or she intake plant protein food or any kind of protein food?
As I was explaining, its specific proteins are responsible for allergenicity. So, if the person is allergenic to beta-conglycinin and that person is allergic to plant protein soy proteins now for example, it is very specific to specific proteins and specific human being. Allergenicity cannot be generalized. I am not allergic to soy protein, I can tell you directly. I can eat soy protein without any problem. Not my friend, he is allergic to soy protein. Now I can't generalize, say, oh, shellfish is allergic for you, now you eat more plant proteins. Maybe that person is also allergic to plant protein, I have no idea. So it has to be tested person by person wise. Did I answer your question? There is a big list of allergens here. I really recommend go and read this paper. They have made a big 4 big tables to showcase how many allergens you have in plant based. I do not say that do not eat allergens, do not eat plant based, that is a very bad idea, because as I said, he might be okay with cereals, but he might not be, it is very individual, okay. And he might be allergic to if he eats soy flour bread, but he might not be allergic if the same bread is prepared by soy protein isolate. Am I clear? There is a lot of processing involved that can change the allergenicity, okay. I have some examples, I do not cover everything, just to show you that we can use amino based, ELISA based method to evaluate allergenicity. When you have time, just go through it. Now I will move on to the next interesting topic called anti-nutritional factor. I am pretty sure I will cover it by 45 and you can go and have your lunch. Is it okay? Just bear with me until I cover anti-nutritional factor. Anti-nutritional factor, as the word says, they are the components, they compromise the nutritional value, okay. They are the components, they compromise the nutritional value of protein in this particular case, okay. Protein has to be digested, but sometimes they become not digested because you have an anti-nutrition factor associated with them. There is a lot of anti-nutritional factor. Okay, skip this question, I forgot about this question. We all know that we can't use cereals directly, like let us say soybean, I already asked this question, we need to cook it, right. We can't eat, some people can't eat every nuts, they need to be roasted, right, because of cooking, roasting, germination, we can actually degrade this anti-nutritional factor, then you can consume them the food, right. There is a lot of anti-nutritional factor associated with proteins, they are listed here, lectins, phenolics, phytic acid, protease inhibitors, saponins, tannins and so on, and I am going to cover some of them very briefly. Is it okay? Did you understand what is anti-nutritional factor? They are the one compromise the digestibility or the nutritional value of proteins, okay. Just to give one example, soybean seeds, it contains lot of anti-nutritional factor. Can you see? Soybean seed contains lot of anti-nutritional factor. To eat soybean, either you have to cook it or you have to process it. By processing and cooking, you can degrade most of them. Now, how do we get soybean? I just was mentioning this slide. We do it by dry fractionation or wet fractionation. During fractionation, the anti-nutrition factor is also fractionated, okay. Sometimes they come along with protein, sometimes they go along with the carbohydrate. So, if you are doing wet extraction, last time I was talking about wet extraction, you took the flour, you add salt containing water, you add sodium hydroxide containing water, right, then you get lot of fractions, okay. We say that this fraction containing 70% protein, but you are also fractionating anti-nutrient in the fractions. So, do not be fooled that fractionation will always help you to get only proteins, you are also fractionating anti-nutrients. So, now the protein containing 70% protein, maybe it contains 10% anti-nutrients. So, whenever you do the extraction studies, be careful where you are fractionating the anti-nutrients also. Am I clear? So, based on the type of effect they pose on your body or especially on the proteins, you have two classes of anti-nutrients. protease inhibitors, and then we have other things called tannins, phytic acid and lectins. I will briefly cover some of these anti-nutrients. Please note that there is a lot of anti-nutrition factors for minerals, vitamins and so on, but I am mainly focusing on the proteins. Okay. The first one is trypsin inhibitor. In the stomach, trypsin is an important enzyme, right, not in stomach, in the small intestine, that is involved in the digestion of proteins. Do you agree? In the small intestine we have trypsin, it is an important enzyme involved in the digestion of proteins. Now, plants have some kind of trypsin inhibitors, they are also proteins basically. There are two classes of trypsin inhibitors in soy, well characterized. They call it as STI, Kunitz type soybean trypsin inhibitor, and Bowman-Birk inhibitor. Basically, these are the scientists, they have identified them. Two types of trypsin inhibitors are there. What they do is, they have a trypsin, and this trypsin inhibitor will go and bind to the trypsin, create a very strong complex. Now, what is the job of trypsin? To digest the protein. So, it wanted to go and attack the protein and digest it. Now, it can't do that job. Why? A trypsin inhibitor is binding and interacting it with this, so it cannot go and attack the protein. So, it inhibits the digestion, so that is the problem. So, this trypsin inhibitor can come along with the purified fraction of your protein. This trypsin inhibitor can already present in cooked soybean. So, that is the reason why people say plant proteins are generally less digestible, because they do have some trypsin inhibitors. Am I clear? The job of trypsin is to digest protein, but trypsin is now inhibited by a trypsin inhibitor, which is another protein, which is less digestible, plus it will not allow trypsin to attack the protein. Am I clear? Any question? And the trypsin inhibitor content, there is a table here. We have about 43 to 84 trypsin inhibitor units per milligram of sample here, and you can see that with compared to soybean, some other proteins have less trypsin inhibitor. Can you see? When you say soybean is 100%, you can use black gram P. P contains very small amount of trypsin inhibitor compared to soybean. So, that is the reason why we need to use a variety of different foods. Soy may not be sustainable, plus it also have a lot of anti-nutrients, especially inhibitors. Either you remove it, if you do not remove it, you have a problem. Okay. I do not cover this trypsin inhibitor assay, it is basically there is a lot of methods to analyze trypsin inhibitor activity, enzymatic assays, it is very easy. I will leave the slides for you with the citation, you can look into how to do this experiment. Okay. Other important anti-nutrient present in the plant is phytic acid. The phytic acid is basically a chemical that binds to metal ions. Okay. It chelates, let us say iron, it can chelate zinc, right. Iron, zinc and everything is important for our body. Do you agree? Now, if you have a phytic acid, it will bind to the iron that is important for our body. Now, the iron is not bioavailable, so you might get iron deficiency, and it is one of the problems of phytic acid, right. And phytic acid can also bind to the protein and make insoluble protein complexes. Now, the protein becomes less digestible. Am I clear? Phytic acid can do two jobs. One, it can bind to the metal ions and makes metal ions less bioavailable, and phytic acid can also interact with proteins and makes protein less digestible, because actually they compromise the protein solubility. So, there are a lot of methods to assess phytic acid. In our laboratory we use 3 methods, and we are developing the chromatographic method also. I will briefly explain you how we measure the phytic acid content, only one I will explain. The phytic acid sample we take, this is a sample containing phytic acid. Okay. We add an enzyme called phytase. The phytase will hydrolyze phytic acid into myoinositol. Then we add another enzyme called alkaline phosphatase, phytic acid will hydrolyze into myo-inositol phosphate. Then we add another enzyme called alkaline phosphatase, and that will convert phytic acid into phosphorus inositol, and that phosphorus is measured indirectly using molybdophosphoric acid, and basically you get a blue color, right. It is an indirect estimation. We convert phytic acid into phosphate, and the phosphate is measured in the colorimetric assay, and the blue color intensity is directly proportional to amount of phytic acid. This is the very common assay we use in our laboratory today. Okay. We also have, this is not important, we also have another assay is called ferric ion assay. In this case, we take the sample containing phytic acid. We know that phytic acid binds to iron. Okay. So, we add an iron excess in this one. If there is a more and more phytic acid is present, it will bind to more and more iron 3 we have added. Okay. And the remaining iron 3, which is not bound to phytic acid, is measured using this bipyridine, and it gives I think a colored complex called yellow color, if I am not wrong. This is yellow colored, and that can be indirectly measured. I am not going in detail, it is not important. I just want to tell you that there are different methods available to measure phytic acid. One is Megazyme kit, yeah. What is the color? Pink, okay. I do not recall pink. Have you done this one? It is a pink color. So, basically the idea is nothing but you take phytic acid containing solution and add excess ferric chloride. The ferric chloride will react with phytic acid. The remaining ferric ion will be reacting with bipyridine, and that will get you the pink color complex that can be indirectly measured, right. Yeah. Yeah, that's you have to do it. Yeah, I'm trying to make it more simplified explanation. So I'm sorry about it. Yeah,
it won't kill it Fe3.
Exactly.
And the color is the peach color.
What is the color?
522 nanometer.
Okay, what is the color?
Peach.
Birch. Ah, peach color. Okay. I haven't done this experiment myself. I can say that. I'm sorry. I know there is some clever people here, but I'm trying to simplify things very simple, and I'm missing some things. Don't mind. But I can explain this in detail in half an hour. But the idea here is that, as I said, you add iron 3 in excess, and iron 3 will be reacting it with phytic acid, and the remaining iron 3 will be reduced to iron 2 by this thio compound, because this is a reducing agent. Then you get iron 2. The iron 2 will be selectively reacted with bipyridine, and that will be measured. I hope my explanation is correct now. And that takes me extra 5 minutes, so I really simplified my explanation. I skipped some information, I am sorry about it. Is that clear? Any question? It has its own advantages and disadvantages that I will skip it. So, you can use these two methods to quantify phytic acid. And then we have another anti-nutrient called lectins. Lectins are nothing but carbohydrate binding proteins. They love carbohydrates. Okay? Lectins loves carbohydrate. What they do here is that in the body we have proteins, and they are connected to sugar. Okay? In the body we have proteins, they are connected to sugars, and we call it as glycoproteins. Okay? And lectins can also like glycoproteins, and they start interacting it with the glycoproteins, and they can compromise the digestibility. Okay? Glycoproteins have a huge role, I do not go in detail today. So, they compromise digestibility, especially in the small intestine. Okay? That is brief explanation. Just remember that lectins are carbohydrate binding proteins. They can interact with the glycoproteins in the body and compromise the digestibility. And here is the table that shows how much lectins we have in plant based. 2.5%, 3%, 1%, a lot of lectins are present. But most of these lectins are purified when you do the isolate preparation and concentrate preparation. So, they are gone. But when you eat a whole seed, there is a lot of lectins. Am I clear? Then we have a glucosinolates. These are very characteristics to this Brassicaceae family, for example, mustard or rapeseed, and they are all containing glucosinolates. And the glucosinolates are actually good in some senses, in some cases they are bad because there is an enzyme called myrosinase, and that can cleave glucosinolates and convert glucosinolates into thiocyanate compounds. They can cleave this moieties. And these thiocyanates or isothiocyanates are not good for the body because there is a lot of theories. They say that they can interact with the hemoglobin and can avoid the oxygen carrying capacity of hemoglobin. There is a lot of problems. So, they are considered as anti-nutrients. They can come along with the proteins. So, here is the big table showing glucosinolates content in different in rapeseed, different glucose. There is a lot of 100 different type of glucosinolates. They are varying in the different concentration, especially in the rapeseed. So, we can identify and quantify them. Then I will come back to the tannins and phenolic compounds. They are also anti-nutrients. This one I have already explained in my 2 lessons before. You remember plant proteins are colored, okay, and because of the phenolic compound, polyphenols, okay. Some people call phenolics are very healthy, bioactive. Lot of people are doing antioxidant activity of phenolic compound, antioxidant activity of anthocyanins. The literature is kind of biased here. Some people call it as anti-nutrients, some people call it as good bioactive. There we actually debate each other. I do not know which one is right here. On a protein context, okay, on a protein context, they are anti-nutrients. Exactly. Again, it is a debate thing. I read 100, there is a 2000, 20,000 papers on phenolics and antioxidants. Everybody does. Total phenolic assay is the most common assay done by all the bachelor's, master's and PhD students. TPC analysis, they love doing that, easy. Total flavonoid assay, very easy to do. We do everything. Oh, my extract is antioxidant. Wow, 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 super. Let us not go back to that story. It is nice to do, but it is some glitchy thing. Okay. I am not going to all these things at the interest of time here. So, phenolic compounds, what they do is, this is protein, right. I am eating this one, I need to digest it in my stomach, right. Now, these proteins come along with the phenolic compounds, right. Now, the phenolic compound I told you in my previous lesson, I gave a lot of explanation. They attack to the protein. They can interact with the protein covalently and non-covalently. They can make a protein phenolic compound complex. Now, this complex might not be digestible in our body because enzymes are very specific to peptide bonds, right. I taught you that one. Our proteases are very specific to peptide bonds. Now, there is a peptide bond here, C double bond O NH, okay. Next to this peptide bond, I have a phenolic group attached, a very big phenolic compound attached here, this one, okay. Now, my enzyme is coming, it wants to attack this peptide bond. What it will happen? It cannot access it because enzyme should go and sit on the substrate to cleave, right. Now, we cannot access it because you have a big phenolic group attached like that one. Am I clear? Now, the accessibility is a problem, so the digestion can be compromised. Not only that, these phenolic compounds directly sit on the enzymes because enzymes are also protein. So, it will inactivate the enzymes. So, you compromise the digestion. So, 2 pathways. Number 1, enzymes cannot access the protein because of the bulkiness of the phenolic compound. The second one is phenolic compound directly interact with the enzymes, can inactivate enzymes, it can bind to its active sites, then enzymes of no use. So, it will compromise the digestibility. Is it clear? So, it is an anti-nutrient on a nutritional point of view, not a bioactivity point of view. That is what Asta was telling, right. For proteins, it is an anti-nutrient. Here is a table that shows different amount of anti-nutrient tannins in different foods. I do not read the numbers here. When you are free, just go and see, just to let you know that plant based proteins can come up with a lot of phenolic compounds. Okay. This is a summary table, it is also for the homework. this table summarizes different anti-nutrients compounds and how they act and so on. Now, in 5 minutes I will talk about how do we eliminate anti-nutritional factors. I will not go in detail, it is not the scope of today's lecture. You can do too many things to kill the anti-nutrients. You can do germination, you can do soaking, you can do heat treatment, you can do fermentation, lot of things you can do to remove anti-nutritional factor. Because imagine, just to give one example, trypsin inhibitor is this is trypsin inhibitor, okay. Trypsin inhibitor, it is a protein, do you agree? It is a protein. Now, if I add an enzyme, if it will cleave the trypsin inhibitor, now trypsin inhibitor is not active any longer. Am I clear? If you heat it, the food if you heat it, trypsin inhibitor can be inactivated because it will lose the 3-dimensional structure. Now, it is no longer an active enzyme. Am I clear? So, you can do heating, but every method cannot be used for everything, because if I heat it, you will also denature your protein, okay. You will kill the trypsin inhibitor, that is right, but you will also denature the protein. So, you need to find out the right method for right sample at the right time to get the right benefit. Am I clear? So, there is no universal method to remove all the anti-nutritional factor. You need to try with so many things, and one of the things will work. We are not talking about removal of anti-nutrient factor 100%. We are talking about reduction, that is enough for us, okay. To a small level, anti-nutritional factor does not have any problem to your body. It is only they are present in a high quantity, they compromise the digestibility. Am I clear? So, this is the last slide of today. Coming back to Asta's point, sometimes we call it as anti-nutrients, sometimes they are good. For example, phytic acid is an anti-nutrient for proteins and minerals, but it could also be healthy because minerals are sometimes problematic. Some minerals have to be chelated, then phytic acid will do that job. Phytic acid is a fantastic antioxidant. Anti-nutrients, antioxidants, they are different, but phytic acid is doing both the job, right. Polyphenols, and she says if you drink a glass of green tea, everybody says, oh, drink green tea is good for your health, right. I am telling it is an anti-nutrient. It is a good antioxidant, it can do cardiovascular prevention effect, it has anti-diabetic, every property is associated with polyphenols, but for proteins it is anti-nutrient. So, what I wanted to tell you here is that do not take the term anti-nutritional factor so seriously on a generic word. It is anti-nutritional factor on a protein world, okay. Their effect is depend on how much you eat and together with what you eat. Maybe it is a bad idea to drink green tea together with a particular protein. Maybe it is a bad idea to add milk to green tea and drink it. Maybe both are not bioavailable now. I don't know. I am telling about the research we are doing in our laboratory, right. So, it is more about how you are mixing two things, to what extent we are eating, and that determines their anti-nutritional value or bioactive value. We as a food scientist have to be very careful about using the terminologies. So, that brings to the end of my lecture. I do not recall the number it is more about allergenicity and anti-nutritional factor. So, to sum up, what did we study today? We studied from the protein quality, yeah, bioavailability, digestibility, yeah. You can give her a mic, yeah. Asta tried to summarize today's class. Stand up and summarize. Yeah.
Okay.
Yeah.
So first we talked about how to evaluate the protein quality, because when we are taking the alternative proteins, we need to focus on the protein quality as well.
Not only quantity, is quality matters. Yeah.
And that can be determined in two ways.
Yes.
First is digestibility, and in other terms we can say it bioavailability as well.
Yeah.
And about the amino acid composition analysis.
Absolutely.
So we talked about how we can determine these both, right? And bioavailability and digestibility can be performed in two ways using two models, in vivo and in vitro.
Yes.
In vitro, that we carry out the analysis in test tubes.
Yes.
Right, and
infogest
model.
Yes.
And they have two basically models like static and dynamic. Dynamic, we have quite modern instruments which mimic the stomach effect.
They mimic the stomach effect.
And in static model we have infogest. And in vitro we perform in two ways, PDCAS.
In vivo we perform in two ways.
PDCAS and DIAAS, right. So PDCAS is performed basically in mouse and
in rats.
Right. And DIAAS is performed using pigs model. Right.
And we believe that DIAAS is better than PDCAS.
Right. Then we talked about the amino acid composition analysis, because essential amino acids are very much important. So we have to analyze what is the composition of that protein in terms of amino acid. So we utilize, first we have to break down that protein into smaller components, that is amino acid. Then we can use GC, FID, and UHLC, right, with the FI detector.
Yes, fluorescence detector.
And then we talked about the allergenicity.
Yes.
And the anti-nutritional factor. In the allergenicity, we know that there are typically all over the world 8 major allergens are present. And our body recognizes some protein as antigen.
Yes.
Means our antibodies think that they are bad for our body. So our innate immune system activate, and they give symptoms in the form of rashes, respiratory effect, or indigestion.
Yes.
So and after that we studied about how to eliminate those allergen or how to detect those allergen. We have ELISA method and skin allergy detection test also. And after that we talked about the this anti-nutritional factor. The major anti-nutritional factors that we talked about was directly they are binding the protein. They are making the protein unavailable for the human body.
Exactly.
So major were lectins, trypsin inhibitors, and
phytic
acid,
glucosinolates,
tannins. So they do have the negative effect on the body, but they have few health benefits on the body, but still we have to remove so that we can get the maximum of the protein. So we talked just summarize the methods to eliminate those anti-nutritional factor as well. The physical and biological method, we can do it by soaking or heating, and in terms of biological treatment we do it with germination or fermentation or other enzymatic treatment.
Absolutely, that is a fantastic summary, Asta, and claps for Asta. So well done, thank you so much. And I don't want to summarize again, you have done my job. Thank you so much. And that's it for today, and thank you for attending today's class. And I was so efficient, I only took 15 minutes extra. Yeah, you have a question? Okay.
Sir, coming back to first lecture, really these plant-based proteins replace animals, as there are number of challenges like anti-nutritional, sustainability issue, toxicity, nutritional deficiency, functional, techno-functional, they are lacking techno-functionality, they are lacking some essential amino acids, bioavailability is less. And when we consider all these lectures, to conclude one, are they really as compared to plant proteins, animal proteins, and as we need more, enough this processing modifications to get the desired as we are having already in animal form?
Very good question. It's a matter, it's a debate question basically, right? It's a question for debate. He's asking, Mahesha, you're telling too many things that we are doing to get the final product. It is digestible, less allergenic, and no anti-nutritional factor, and you extract from so many things. What the heck is this? So many things we need to do here. Oh my God, is it really good? And that's his question. Is it really replacing animals? There is a matter, I think we can take it in one of our tutorials, this question actually, that you all come up with your own opinion. Please note that the driving force here, politically and scientifically, is that we are 100% sure that alternative proteins are better than animal proteins in terms of environmental footprint. There is no doubt about it. There is no doubt about it. Environment is the most important thing, we have to keep it in mind before digestibility, right? because if there is a greenhouse gas, even if you do not eat protein, probably our offspring cannot survive in this environment. That is a fundamental thing, right. That is the biggest thing we have to solve today compared to other things. And we know that food science and technology, or food scientists know, food systems in general is responsible for 1/3 of global greenhouse gas emission. Take that point into account. And that is the reason why, although we have a lot of problems with plant based world, solubility, digestibility, anti-nutrients, allergenicity, we have to work on this one. We have to do a lot of science and technology here to get the best out of plant proteins. We are doing research. I am not telling that they are the best already or they are the worst already. No, we are doing the research and development. I am pretty sure maybe next 5-10 years we will solve all these problems. We get the best protein out of plants or insects or microalgae. That is the only answer I can give you for now. It is a matter of debate, and I will give you opportunity for every single of you in one of the afternoon session that you can put your own opinion. Okay?
So I also have to add on this.
Yes, Asta wants to add on this one. Yeah.
The answer to his question was already discussed in the first lecture. That we started with why we have to study this alternative proteins.
Yes.
And in that sustainability, many of the points were elaborated in a very well mannered. But what I think a food technologist or food scientist in true term has to innovate to the next level so that our upcoming generations and the conditions, different different occupations have different conditions as well. Like if we have to fill up the protein dietary allowance of an astronaut, then we can't send the meat over there.
Absolutely.
Right? So we have to work on that alternative proteins which have the better shelf life, which have the better digestibility, which have the better bioavailability with the least anti-nutritional factor, and we can manufacture and send to them so that they can survive over there. And same goes with the army personnel as well.
Absolutely.
Right? So that's why we keep on working, and the food technologists have a lot of things to give to the human population.
Absolutely, we are progressive, we are not traditional. That is one thing I have to mention, that we have to be progressive people. We can't stick to what my grandparent has eaten continuously. I know my grandparent has eating a very sustainable diet, but things are changing now. So we need to change things. We need to be a little bit progressive. Lot of things she has mentioned, that we have different commodities here. We are not just scientists sitting in the lab. We need to feed army people, we need to feed astronauts, we need to feed vulnerable people. For example, young children, they have no access to breast milk because things are changing. We need to create new infant formula. We have old people, they are not getting protein that is digestible. We need to create alternative proteins for them.
WHO with FAO is working on this project also. In many of the Asian and African countries where the people are suffering from the malnutrition. So the packets are being formulated with the dietary allowance in terms of the protein, so that they won't have the deficiency, kwashiorkor and marasmus in early childhood.
Excellent. So there is a lot of need actually, right? So
and plant-based proteins are only utilized for that.
Exactly, and they're sustainable. That's one reason. But there is the processing is also important because you need to supply for a long distance. I know I'm eating some people's lunch break because at hostel food will be closed at 1:30. So one last question and then I have to leave. Okay? Yeah.
Sir, we are putting so much of efforts in plant proteins. Instead of that, insects are readily available. They are easy to produce, and we can get more high quality protein. In terms of quality, plant based are very less. In terms of bioavailability, quality, insects might be good. Why do not we shift to insect proteins?
That's again, I have already discussed that in one of the lectures, right? I have clearly told insects are the best in the world, but consumer acceptability is not best good for them. It's more about changing the consumer feeling also, right? And that takes a lot of efforts. I'll discuss that in detail in one of my future lectures. How do we turn consumers' mindset? It's not about me as a scientist telling you eat this one. Will you eat that one? My mother will not eat if I say eat this one because it's best. She has her own perception, right? Why do I care for you because you say it's good, I don't believe it's good. I want to eat what I like. It's personal, right?
Absolutely.
And there all these things matters. We will take it in the next future classes. I really have to stop here because I know people will be unhappy otherwise. And thank you for today. So we had a fantastic session, and tomorrow we discuss more about other topics about chemical modification to answer your question, how do we create high quality proteins. Thank you so much for today. That's it. We end.